Happy New Year and welcome to the 2022 Telomere Summit. My name is Eric McDonald. I'm the Director of Sales at Telomere Activation Sciences, or as many of you know, us as TA Sciences. We are proud to be celebrating our 20th anniversary as a company, and we wanted to thank the thousands of physicians and their respected patients in believing in our product, TA65. The impact this molecule has had is utterly remarkable. We were fortunate enough at the beginning of 2021 to publish our 10th peer-reviewed clinical trial to date. This randomized double-blind placebo-controlled trial of 500 patients was looking at the impact on the immune system and the immune biomarkers. Many of you know our impact to date with activation of telomerase in lengthening telomeres, the end caps to the chromosomes. This study is looking at the impact on the immune system. The main takeaway is that we saw a 21% reduction in senescent T cells and a 12% increase in naive T cells. As many of you know, as we age, we tend to accumulate senescent T cells, especially in the population that suffers from cytomegalovirus. As well, naive T cells tend to decline with age. So to see a product that can actually lengthen, or in this case, increase naive T cells, again, is utterly remarkable. I'd like to thank our many sponsors, our global expert speakers, and especially our host, Dr. Joseph Raphael. I truly believe this unique event will open your eyes and especially your mind to the unbelievable science of telomere biology. After the event, feel free to contact myself or my colleague, Sebastian Conte. If you wanted to just pick up the phone and give us a call, you can call our headquarters in New York City at 212-588-8805. I'd like to also acknowledge our founder and chairman, Noel Patton. Without Noel's support and driving force to make TA65 the molecule that has had this tremendous impact on thousands upon thousands of human beings across the world, we wouldn't have this opportunity to share with you this 7-day Telomere Summit. So with no further ado, I hope you truly enjoy the show. Thank you and have a wonderful 2022.
Hello, and welcome to the premiere day of the 2022 Telomere Summit. I'm your host, Dr. Joseph Raphael, and I'm so excited to be here with you today. In our first episode, we'll talk to Dr. Bill Andrews, arguably one of the premier telomere biologists in the world, about the history of telomere biology, its role in degenerative diseases such as cardiovascular disease and cancer, and why he thinks that lengthening telomeres could well be a cure for aging. We'll then move on to talk to Dr. Gordan Lauc about the newest biomarker of aging on the block called the GlycanAge test that he has developed, and why glycobiology is important for understanding inflammaging, and in particular, the effect of hormone replacement therapy on aging as a new molecular marker to tell you that your HRT levels are in good range. Then we'll talk to Dr. Bill Pawluk about pulsed electromagnetic field therapy, its effect on multiple medical conditions, and how it affects cellular aging and health. We'll then move on to Dr. Chris Shade, who will talk to us about detoxification, its role in improved cellular signaling and telomere length maintenance. And finally, we'll wrap up the day talking to Dr. Robby Besner about harnessing the sun's infrared energy for natural anti-aging and life extension through photobiomodulation. For many of you interested in telomere biology, Dr. Bill Andrews needs no introduction. His vast knowledge of telomere biology requires that I interview him in two parts. He is the founder and CEO of Sierra Sciences, a company focused on finding ways to extend human lifespan and health span through telomere maintenance. As a scientist, athlete, and executive, Bill continually pushes the envelope and challenges convention. He has been featured in Popular Science, The Today Show, and numerous documentaries on the topic of life extension, including, most recently, the movie The Immortalists. Bill has been a medical researcher in biotech since 1981, focusing on cancer, heart disease, and inflammation research, though his passion has always been aging. In the early to mid 1990s, while at Geron Corporation, Bill led the research to discover both the RNA and protein components of the human enzyme called telomerase. Bill earned his PhD in molecular and population genetics at the University of Georgia in 1981. He then served as senior scientist at Armos Corporation and Codon Corporation, director of molecular biology at Burlex Sciences and at Geron Corporation, and director of technology development at EOS Biosciences before starting Sierra Sciences in 1999, where he now serves as president and CEO as well as chief scientific officer. Bill is also a named inventor on over 50 US issued patents on telomerase and author of numerous scientific research studies published in peer-reviewed scientific journals. Bill is also an avid ultramarathon runner. He regularly competes in 50 and 100+ mile races, often finishing at the top of his age group. He considers endurance exercise, when done properly, to be a way to significantly slow down the aging process. Bill's obsession is to cure aging, and that includes his own aging. His regimen to slow down his own aging and declining health is unique and based on years of his own research. Well, Bill, it is fantastic to have you on the Telomere Summit. I've been waiting to interview you because, of course, talking about telomere biology without talking to Bill Andrews is, it's just, that's not something you can do. So you've been in the field for decades, and I'd love to have you start by just giving the audience a little bit of perspective about that you kind of uniquely have on where, why you got interested in telomere biology, what you've been doing with it, and where a little bit you think things are going in the future.
Well, I've been in telomere biology for 40 years. I guess I started in 1992. So it's, 40, almost 40 years, 39 years, 40. And it mostly, I got into telomere biology because of previous thoughts on what aging is and stuff like that. And I kind of like always thought aging had to do something to do with the Hayflick limit. Leonard Hayflick has shown that our cells can only divide a certain number of times and then they stop. And before I knew about the Hayflick limit, it used to always bother me that, we're made up of cells, and when cells get old and unhealthy, why isn't that other cells don't divide and replace those cells? Just like bacteria in a petri dish, if some bacteria get sick or unhealthy or something like that, they just disappear because the healthy bacteria outgrow. Well, that might happen to us in our younger years, but why doesn't that happen to us when we get older? And so that was always a bother me. I mean, I'm not talking about right now about the cells that don't divide, like our nerve cells, but those are also like, they have cells that take care of them. I call them caregiver cells, right. like glia cells and Schwann cells that take care of neurons. Those do divide, okay, a lot. So the division is still involved even in the non-mitotic cells. But why is it that we don't, we know that our cells are dividing? Why is it that they get old? There had to be something going on, because a bacteria culture doesn't get old. Why do human cells get old? So then I learned about the Hayflick limit, and I guess Leonard Hayflick learned about it in 1961, but I didn't hear about it until I would say early mid 1970s. And at that point, and actually, so it was early 1970s, because I know that when I started applying to graduate schools and stuff, I started saying that I want to go someplace that's looking at what's causing the Hayflick limit. So, because I kind of felt that had something to do with aging. And so what would cause something so specific to happen? There had to be something like almost a program in our cells that told our cells to stop dividing. I kept it coming up with the analogy. It was like ride tickets at an amusement park. Okay, every time our cells divide, you lose a ticket, and you only have a certain number of tickets. So I'm just struggling with this year after year. I'm talking to people like Dr. Mike Rose and Sabi Harvey Sarbor about starting companies and stuff like that back in the late 80s, early 90s, about focusing on trying to find out what's causing this Hayflick limit. And then just one day I'm sitting in a conference in Lake Tahoe, and a guy named Calvin Harley is getting up on stage to talk about telomeres, and I had no idea. I knew what telomeres were because of my background in biology and stuff like that, but I had no idea that he was going to say what he did. And that's what he said, that telomeres shorten as you get older, and that you can actually measure the length of a telomere and you can tell how old a person is. And more importantly, you can guess how long it would be before they die of old age. Well, I'm jumping up and down in my seat because I thought, my God, these could be those ride tickets.
Right.
And they're the only thing that I ever heard of that could be these ride tickets. Everything else, every theory about aging did not have the twos all adding up to something that made sense about aging. So this telomere stuff sounded fantastic. And as soon as Calvin got done, I was at the bottom of the podium as he was coming down the stairs before he even got off the stage, just saying, I want to work with you. You know, I want to get involved in this. And I literally jumped ship. I was working with a company called Burlux Biosciences, and I used to call myself a lifer. I was never going to leave that company. I loved it so much. But all of a sudden, here I am getting ready to go work with Calvin Harley. And so it was literally 3 months later that I was actually working with Calvin at Geron Corporation as the director of molecular biology. That was, I think, 1992, might have been 1993. But that's when my career in telomere biology occurred. And I just became obsessed with it because suddenly I found these ride tickets. And so I wanted, and the way to show that the ride tickets were causing the aging was to figure out a way to add more ride tickets back and see if they gave you more rides or more cell divisions. So I became just obsessed with that. I literally, Geron Corporation had to build bring in a trailer with beds so that I could sleep there because I was working there around the clock.
Wow.
But you know, and there were other people working on trying to discover the enzyme telomerase at the time. And you know, suddenly I found myself working with them, collaborating with them and stuff. But I felt like everybody was making too many assumptions and going the wrong way. So I started a whole program at Geron Corporation. Geron put 3/4 of the employees all working for me. So we started a program to start working on some other approaches that I thought made more sense. And lo and behold, 3 months later, we were able to discover the RNA component of human telomerase, which didn't have any similarity at all to the previous one that had been discovered in Tetrahymena. And that was one of the mistakes that other people were making, is they were expecting it to be similar. And so they thought they could identify the human one by sequence identity to the human, but it wasn't. So we ended up discovering it through probably one of the toughest research projects I've ever been involved in of brute force, but we ended up discovering it. And then people still wouldn't believe we discovered it. And to prove that we had discovered it, we mutated the sequence in the RNA component so that instead of encoding the normal TTA-GGG, TTA-GGG, TTA-GGG repeats, we mutated it so it now would produce TT-GGGG, TT-GGGG, or TTT-GGG, TTT-GGG. And so we made these things and put these RNA components in the cells, and then isolated the telomeres and sequenced them, and lo and behold, they were the sequence we'd wanted them to be. So that proved that we had discovered the RNA component of telomerase. And then that shortly thereafter led us to the discovery of the protein component of telomerase. Very first thing we did was, okay, let's see if this abolishes the Hayflick limit. And lo and behold, it did. Now, at the same time, we did an experiment where we took the antisense of telomerase and put it into cancer cells, and we showed that it killed every cancer cell. So it was almost in the exact same week that we'd come up with a potential cure for cancer and a potential cure for aging at the same time.
Wow.
That was exciting, but
Heady times.
The frustrating part of it was that all biotech companies are business first, science second.
Right.
And so there was a big meeting. All the investors came to a big meeting. We had a big board meeting with all the investors. I gave a presentation. I discussed those things, and I'll never forget the head of the head of the board of directors, chairman of the board, said, "Well, we can't do both. We've got a cure for aging and a cure for cancer. We can't do both. We have to pick one. Which one gives the quickest return on investment?"
Obviously.
Well, even though geron was short for gerontology, aging, they chose to go for the cancer. Now, I had previously had many discussions with Calvin Harley and others saying, because of my background in cancer was, I'd already been awarded the second place for National Inventor of the Year for my cancer research. I had been involved in a lot of cancer research prior to going to Geron Corporation. So I knew a lot about cancer. And I was saying that inhibiting telomerase with an antisense isn't going to cure cancer. It's going to make it go away, but it's going to cause telomeres to get shorter. And we already knew that mutation rates skyrocket when telomeres get shorter. And that inhibiting telomerase was just going to make telomeres get really shorter. Mutations rates were going to skyrocket. And some of these mutations were going to convert those cells into ways to survive any cancer treatments, or in fact, start producing their own telomerase. And so I was pretty upset when they decided to focus on that. I am the real inventor of a meltastat. I'm not even certain I've pronounced it correctly, which is the antisense that's in clinical studies now for Geron Corporation that they use to inhibit telomerase. And I would always say that the cancers are always going to come back. And then they're going to be resistant to the meltastat and resistant to whatever you treat them with.
But if it gives 6 months, it'll get approved.
Yeah, well, I mean, it is anything that helps. I strongly agree that they should continue pushing it, because it very effectively makes the cancer go away. But people always have to be aware of the fact that it's going to come back. So people do live longer because of it. But it's like, it's not really the cure for cancer. And we do have a cancer program here at Sierra Sciences, but we're trying to focus on not inhibiting telomerase. We're trying to poison cells that produce telomerase. Okay, so instead that keeps the telomeres long. In fact, it's a two-hit kind of thing where we first, even if it's a cancer cell, we induce telomerase to lengthen telomeres, and then take the telomerase inducer away so that all the other cells in your body shut the telomerase gene down, and then add a poison. We have two different types of poison. One is a nucleotide analog that incorporates into the telomere that will actually kill the cell that other polymerases won't recognize. And the other one's using the same approach that we used to prove that we have telomerase back in it was to make a mutation to the RNA and infect cells with a gene therapy that delivers this mutated RNA to actually produce a mutant telomere that would kill cancer cells. And we've even though the ones we originally used, the ones that converted TTA-GGG to TT-GGGG or TTT-GGG, we've done a lot of work on like all the different 5,000 plus different combinations of telomere sequence, and have found some that are very, very lethal to cells. Oh. The problem is delivering the gene therapy to all the cells is the issue. But the nucleotide analog, which we don't have yet, we're still searching for things. Woody Wright and Jerry Shea, or it's Jerry Shea because Woody Wright passed away. Jerry Shea published a paper recently on a nucleotide analog, but it's not sufficient. A nucleotide analog that actually poisons cells, but it's still incorporated. It gets incorporated by other polymerases. So what we're looking for is something like that would be totally ignored by like RNA polymerases and DNA polymerases besides telomerase. Oh. So we do have a cancer program, but the idea is to keep telomeres long, not get telomeres short. So getting back to the subject, as soon as they decided that cancer had a quicker return on investment, despite everything I had said, I resigned. In fact, I stayed there a little while, and I led the first studies that showed that we could overcome the Hayflick limit. When I presented the data at a strategy meeting, I was reminded that I wasn't supposed to be working on that. I was supposed to be working on cancer. And at that point, I kind of like walked to my office and called a venture capital firm that I'm friends with and said, hey, I think I'm looking for something else to do. I want to start my own company now. Is there something I can help you guys with to earn some money while I'm trying to start my new company? And they just had me consult a lot for, go to other companies to salvage their science and things like that. And so that was a win-win situation for me and them. And it allowed me to get time to write a business plan and get investors and start Sierra Sciences. So Sierra Sciences has been focused on trying to find a way to produce telomerase inside of all your cells, despite the fact that some people believe that that's going to cause cancer. I can't really say if it is or isn't. I mean, there's no data yet. Okay, all of it, everything is hearsay so far. There's not like I go to conferences and there's people, scientists and doctors that believe that there's actually been studies showing that when you put the telomerase gene into a cell, it becomes a cancer cell. But there is no data at all saying that at all.
And there's data that shows that when you turn on telomerase in a model that it doesn't necessarily get cancer.
Yeah, actually cancer gets decreased. There's several studies now showing that, surprisingly. In fact, Dr. Rhonda Pennell, remember, he was a cancer, he still is, he's a cancer researcher. When he did his mouse studies, he wasn't trying to show that the mice were going to have a remarkable reversal of the aging process, but which he called it. He was actually trying to show that they were going to get cancer because he was going to try to use it as proof of how important it was to find a way to inhibit it.
So you should probably just explain a little bit about that experiment that was so serendipitously good for the aging research and for telomeres as anti-aging. Just what he did, because I don't think we've talked about that at length, and you're a perfect person to talk about it.
Yeah, well, what Rhonda Pennell, he got the genes from Geron when I was there. Ron and I actually collaborated a lot on trying to figure out whether or not, like Maria Blasco's original data, where she originally announced that telomerase and telomeres have nothing to do with aging, because when she put telomerase into mouse, or when she knocked out the telomerase gene in mice, they still died at the normal age. But mice don't age by telomere shortening; they age by other mechanisms.
Right.
And their telomeres were so long that it had to go. Rhonda Pennell and I worked together to show that it was actually predicted that it would take 6 generations of mice before the telomeres got short enough that they would start showing signs of aging like humans. Then lo and behold, that turned out to be the case. So Ron and I had a good working relationship. Geron Corporation provided him with the tools, but he wanted to show that telomerase causes cancer, and I was all for it. I mean, it's like let's get this done and let's see it. So what he did is he first engineered mice that were knocked out for the telomerase gene, for the mouse telomerase gene, and he then incorporated a new telomerase gene that was under the control of the
tamoxifen.
Well, I was going to say an inhibitor of the estrogen receptor. Okay, so
tamoxifen, yeah,
4-hydroxytamoxifen,
right,
or dihydroxytamoxifen. I can't remember what it was, but so he put it so the mice had telomerase turned off in the absence of the tamoxifen drug, and then when they got old, when the telomeres got really short and they got old, so he let them go to 6 generations to get short telomeres. Then when those 6th generation mice started to show signs of human aging, he then fed them the 4-hydroxytamoxifen, and they got younger by every measurement he could do. Okay, brain sizes got bigger, memory came back, spleen size came back. I can't remember. It's been 10 years now, but all this.
The pictures of the gray coat went nice and glossy brown, and they had good subcutaneous fat. They just looked like younger mice, and on the inside they were younger mice too. Well, it was also, to refresh my memory, was it a 50% knockout so that they only had 50% of telomerase, and then they gave it was a full
knockout? Yes.
Full knockout. Oh, okay.
will knock out. So they had regular telomere shortening. But the when he got interviewed by Barbara Walters on that, he called it a remarkable reversal of the aging process.
Yep.
And I was sitting thinking, that just destroyed his whole theory. I mean, what's he going to do now? Well, he had a very successful career, and as a result, he became the head of MD Anderson in Texas. So he's still focusing on cancer, and there's still benefits in cancer cells inhibiting telomeres to kill the cancer. You just got to do it in a way that doesn't increase the mutation rates at the same time. So I'm hoping that some of our research here will contribute towards Rhonda Pinnell's research.
I think that's what a lot of people, doctors and other healthcare practitioners that think about doing something to a therapy that will increase telomerase activity, they say, well, gee, I mean, what I know about telomeres and telomerase is that, all cancer cells, 95% of them have turned on telomerase, and that's what helps them do the job. So what if we turn on telomerase? Aren't we going to cause a problem with our cancer cells? And, it's on the surface reasonable to think that way, but the details, which you're just about to explain, are that it's more complicated than that.
Well, yeah, the one of the main reasons we get cancer is a weakened immune system, and keeping the telomeres long in our immune cells is going to help our body fight the cancer. But also, one of the main causes of cancers is the short telomeres. So one of the reasons why cancers evolve so late in life is because of the telomeres get shorter and the mutation rates go higher to increase the chances. I mean, people used to talk about accumulation of mutations with age, and you just build up more and more and more mutations. But a lot of studies have come out lately, in the last like 5-10 years, showing that these accumulations don't occur gradually. They occur all of a sudden in the older cells. And so it's probably being induced by the short telomeres. I mean, not saying short telomeres are the only cause of these mutations. Other things can cause them too. But usually when you get a mutation, it causes the cell to be unhealthy in some way or recognizable by the immune system. So either other dividing cells will replace it or the immune system will knock it out until the immune system gets weak because of short telomeres, and then you get immune senescence. Now, this is still theory, because the only way we're going to ever know is to find a way to actually lengthen telomeres. And I'm not talking about just some of the telomeres. Like some great products on the market right now will lengthen telomeres, but they'll lengthen the shortest telomeres. They'll lengthen maybe the average telomeres in some of the cells. But what we need to do is we need to make it so that all the cells in the body have their telomeres lengthened so that they have a young phenotype, and also that they're not causing high mutation rates, to actually see if telomerase is going to cause cancer or prevent cancer. And when I take all the pros and cons and read all the papers and do my critical meta-analysis of the scientific peer-reviewed studies and try to weigh the different things, I come up always with the idea that if I had to bet money on it, I'm going to say that inducing telomerase is going to decrease cancer incidences more than it's going to increase. But it can do both. It's just that in the long end, what caused more cancer? Lack of telomerase or presence of telomerase? And I think it's the lack of telomerase is going to cause more cancer than the presence of telomerase. But we won't know until we can actually test it, and we can't test it until we have a drug or a gene therapy that does lengthen the telomeres in all the cells. And that's what I'm trying to do. I'm trying to come up with something to do, and something like that doesn't exist, in my opinion, even though there's some papers that I wish I would see reproduced to see if they actually can be reproduced.
Right. Yeah. I mean, I think, you're talking about more like a 30% increase in telomere length that would have those kind of super rejuvenating effects, and in every cell. There's been some signals in papers with increase in telomere length, but not like the Stanford paper where they did it with, I think it was an RNAi, or I forget exactly, a messenger RNA, where they lengthened in vitro telomere length by 30%. Then, something like that might be have these incredible effects. What was the lengthening of Depino's telomere length?
30%.
100%.
If you had 30% increase in telomere lengths.
Right, 30%. Right.
I thought that was why you just picked 30%.
Well, no, there was a Stanford study as well, I think, that had somewhere around that length.
In vitro and petri dishes. I mean,
that's what I'm saying. Yeah.
And we've been able to do that for 40 years. In fact, we did RNA back when I was still at Geron. We took telomerase mRNA and put it into cells and showed that would work. But the big problem with the mRNA is the delivery. Delivering it to all the cells is really a big problem. But it's really, there's all kinds of publications coming out where people are saying they've got big new breakthroughs on how to do it. But the bottom line is I just really want to see it get done. I mean, it's like, I'm not trying to get publications myself. I'm not trying to make a lot of money myself. I just want to cure aging. And right now, I think the best way to cure aging is to lengthen telomeres. And I don't want to waste my time on things that really are just going to give me a paper to publish. Because I really think we got to find a drug. We do have a gene therapy that I'm hoping will be effective, but I don't believe that's the end all either, because it still doesn't get delivered to all the cells. I think the best way to actually lengthen telomeres is to find a small molecule drug, which for some reason has a lot of bad feedback on too. People are saying small molecule drugs don't work, but that's because there's been a lot of poorly designed small molecule drugs. There's a lot of small molecule drugs that work really well. I believe that designing a small molecule drug that induces the telomerase gene is a much better way to get delivery of the telomerase to every cell in the body. And so that's my focus.
Do we know, I mean, we've talked about this before, but telomerase is suppressed in most cells in adulthood, even after birth. Do we know, because what's the mechanism? I mean, the mechanism, some of the small molecules is to de-repress the repressor, but I know you've said that, well, I don't know that anybody's ever really shown that. I mean, the reason I'm asking is then if we know what the mechanism is or have an idea, then it could well be that a small molecule will work quite well. Is that an active area of research for people, or are they just looking to do gene therapy?
Yeah, we spent over 10 years just trying to figure out how is the telomerase gene repressed, or is it repressed, or maybe it's just so coiled up in the chromosome that it needs some strong activators. And we came up with a lot of candidates. We found a lot of things that worked in ectopic expression of telomerase or in a test tube, but we could not get any of these things to work in vivo. Okay, and when I say in vivo, I'm talking about even human cells in a petri dish. I like to call that in vivo, even though most people call that in vitro. So we finally, after 10 years, and we'd done just about everything we can, we never published any of our work because our focus wasn't publishing, but a lot of people in the last 20 years, 10, 15 years have published papers reproducing our results, but we know, my team knows that that's not going to get anywhere because it's not going to actually work in a normal human cell in a body. We finally just decided, okay, so we've done everything we can't imagine anything else to try. No, we haven't figured out how the telomerase promoter is regulated. Nobody else has. And I came up with the thought that, well, maybe there's multiple repressors. And when there's multiple repressors, you can't use the standard technologies to de-repress to see that the gene turns on, because you have to de-repress several at the same time. And how do you find several of them? If to find one of them, you have to de-repress all of them. So we decided to do it the hard way. Okay, and that's we're obsessed with this goal. So if we have to do it the hard way, let's go for it. So we developed an assay that everybody else had said was impossible, and that was a high throughput screening assay that used real-time PCR to detect telomerase mRNA. And even though mRNA is so low in even cancer cells, the level of the mRNA for telomerase is so low, it's something like 1 mRNA per cell.
Wow.
So low that PCR was thought not to be practical. But I had some pretty good people here, and we developed a really good, powerful real-time PCR assay that we can do in high throughput. It's still a trade secret. We didn't publish or patent it. Nobody else has figured it out. Nobody else, if they have, they sure aren't using it. But we were able to screen like 500,000 different chemicals, including a lot of natural products in this assay, without knowing what the repressor is, without knowing how the gene is regulated. But we ended up finding 900 different chemicals that actually did turn on the telomerase gene. Not to the level that we see in cancer cells. And we picked HeLa cells as a standard to go with, because HeLa, the ones from Henrietta Lack, those were the cells that we had determined when I was still at Geron Corporation, produced the lowest amount of mRNA, telomerase mRNA, but were still immortal. So we decided, okay, that must be the amount that we need to get to make a normal human, a non-cancer cell immortal. So the highest we've gotten is 16% of that. Okay, so we have gotten chemicals that induce telomerase to that level. We still don't know how they work, okay, but we know that they, we've done a lot of studies on toxicity and things like that, and they're not causing any side effects. Well, some are, but the ones that we're pursuing don't cause any side effects.
What's 16%? You hear about the TRAP assay as what you use to sort of figure out whether something's turning on telomerase or not. You talk about one-fold, two-fold, three-fold increase. A HeLa cell will be how many fold?
Well, you can't, because it's divided by 0. Okay, so when you get really good at measuring telomerase activity, you find out that there really isn't any mRNA or any telomerase activity in like endothelial cells or fibroblast cells at all. And so when we do our PCR tests and stuff like that, we have to go through many CTs and units of measurement in PCR before we actually see anything from a normal human cell. And we just figure that's 0. That means there's 0 mRNA. So we can't really say a fold increase, but we can say how much fold are we away from getting to what HeLa is.
Right. Okay.
So 16% is about what, 1/6 or 1/8 of
right.
what HeLa is. So if we can find some way to increase it 8-fold more, we could. Now, let's go back to this idea that there's multiple repressors. Okay, well, we're planning to now try to do a synergy study. Okay, to try, let's say maybe some of the repressors that we have found attack one of the repressors, some attack another repressor, and some attack a third repressor. And maybe we might find that mixing them will attack all 3 repressors, and we'll get to 100% of HeLa or even higher. That experiment hasn't been done mostly because of the lack of funding. It's but we're geared up to do that. It's just a very expensive experiment to do. Literally when we were identifying the first 900 chemicals before we did the synergy, even get a chance to start talking about doing synergy, we were spending $1 million a month on the screening. Then lots of things happened: the global financial crisis, the Tokyo earthquake, the pandemic, COVID pandemic, all these things started interfering with our ability to be able to do this research. So, but that's still what I'm trying to do, and I'm trying to find some way of bringing in funding to do that without losing control of the science. Okay, one thing I learned when I did have investors, and I used to only own 17% of the company, I found that I had very little control of the science. Not some of the investors were outstanding, but many of them would completely take over our strategy meetings and get our scientists geared towards other things. And then there was even talk when I used to complain about it that they were going to fire me and replace me with somebody else. And it's like that would end my life, because I'm so obsessed with this. And so finally, I was able to buy out all my investors. So I own 100% and at the company now. But I don't want to bring in more investors that are going to have control. So I'm trying to find funding without losing control, but I've got some great opportunities for investors to make a ton of money off of marketing discoveries if they just let me focus on the science. So I'm hoping something will turn up soon, and we can get back onto this. But I think when we do get the funding that we need, we will be able to get a lot of these questions answered. Does telomerase cure aging? Does telomerase cause cancer? All these things we'll be able to answer very straightforwardly. Right now, they're just all hearsay.
People are looking at, I mean, obviously you hear the sort of the thought from the gerontology community, and it's perfectly valid that trying to cure individual diseases will only add about 7 years to the average human lifespan. But if you cure aging, obviously you're going to add decades to it. But there is work going on looking at, well, there's lots and lots of observational studies looking at the role of telomeres, or anyhow, the association of telomere length with cardiovascular disease, dementia, osteoporosis, of course, cancer in the opposite direction, short telomeres increase. And some of that's probably through affecting the aging process, but some of it's maybe more direct to the pathology for those particular diseases. There's some research, a lot of research in cardiovascular disease. What's your thoughts about sort of that area and clinicians trying to keep telomeres longer in their patients to stave off some of these chronic diseases, and particularly Alzheimer's and cardiovascular disease?
Well, I get misunderstood a lot. I think it's because I'm not a semantics expert. But when I say curing aging, I'm talking about curing Alzheimer's, curing cardiovascular disease, curing osteoporosis, curing multiple sclerosis and all the demyelinating disorders and things like that. Because I believe that telomere shortening plays a critical role in almost every disease we've ever heard of. In fact, I am totally unaware of any disease that's ever been discovered that there hasn't been now a scientific peer-reviewed publication showing that telomeres played a role in inducing that disease. So I believe that lengthening telomeres is going to correct many, many diseases. And I especially target Alzheimer's and cardiovascular disease. We have, as I mentioned before, we do have a telomerase gene therapy that we're trying to get clinical studies underway with. I think it's going to have a delivery problem. We're not going to be able to get to all the cells, but it still might help. But the big problem with it is it costs like $1 million to treat one patient once. And even though it's great proof of concept, it's not going to be something that is going to be readily available to everybody. And so it's not my priority. But I do, I do have clinical studies designed, protocols designed, and you're involved in some of them actually, to treat Alzheimer's, that's our main one, to treat cardiomyopathy, critical limb ischemia, demyelinating disorders, idiopathic pulmonary fibrosis, which is actually something you promoted that we focus on, and many other diseases. I mean, the list is mind-boggling, but these things are super expensive, and it's like I am in contact with the FDA and stuff. I'm forgetting the guy's name. There's a guy at the FDA. I want to say Don Fink, but I don't think it's him. He's very on board. But the big problem with aging, and I agree with the FDA totally on this, is that you can't measure it. Okay, and even though people are constantly saying they have markers of aging, they really aren't markers of aging. They're just markers of the markers.
Yeah, I mean, there's the TAME trial that they're looking at right now, and that's sort of not aging per se. It's multi-morbidity markers, clusters of diseases, and when they have onset. But I guess the DNA methylation clocks are kind of interesting as markers, and there's a lot of buzz about them. It is an area I actually did want to talk to you about, which is, for a long time, measuring telomeres was sort of a biomarker that you looked at. And I've been measuring telomeres in my practice for 15 years now. the R squared with aging, with chronological age isn't that great, but then along came these clocks that were the R squared with chronological ages like 0.95, 0.98. Then there was the TRIM trial that was published, where they looked at growth hormone, DHEA, and metformin, very small, but because Steve Horvath's clock was used and they saw like a 2-year reversal of aging, would you expect someone who was treated with, let's say, Ron DePino's mice? I mean, it would have been interesting if they'd used Horvath clock on DePino's mice, and I wish somebody would do that experiment. It wouldn't be that expensive to do, where that would validate that as a marker if you saw a real significant reversal of DNA methylation, because you see that when you do the Yamanaka factors, the epigenetic clocks get reversed as well. Would you say that that's of course, being a clinician, I'd be like, yeah, the lungs got to work better, the muscles got to work better, the brain's got to work better too. But as a short-term marker, would you accept that?
Yeah, no, I openly say that I think DNA methylation of DNA is a better marker of aging than telomere length measurement.
Yeah.
But not only because telomere length measurements are very difficult to do. Okay, I believe that telomere lengths are probably controlling the methylation. Okay, so and one of the studies that I would love to do, and some people have already done some studies that really suggest this is true. That's at least in vitro, in human cells in a petri dish, let telomeres shorten and re-lengthen them, let them shorten and re-lengthen them, and see if that corresponds with the length changing back and forth of the methylation path.
I bet you it will. Yeah.
It might even be better than DNA methylation is the glycosylation.
Gordan Lauc stuff.
Glycosylation. Yeah. What did you call it?
Oh, you're talking about glycosylation. I mean, you're talking about like the glycome, or are you talking about hemoglobin A1C glycosylation?
I'm forgetting the name of the guy. Gordan Lauc.
Gordan Lauc. Yeah. Gordan Lauc stuff. Yeah.
Okay. Yeah. So I think he's brilliant. I think what he's gotten on to is amazing. And I think, again, though, I think that's probably controlled by telomeres. And
that's interesting. Have you talked to Gordan about that?
Oh yeah. Gordan and I have become good friends ever since I first heard him talk, and was just impressed because, what I look at is how good are people at doing critical meta-analysis of scientific peer-reviewed studies and building theories out of that. And Gordan is just amazing at that, and so are a lot of other people. And I kind of like to work with them. That's one of the reasons I like to work with you too. And it's like, and so getting all this stuff put together is I think putting a model together. And a lot of these people agree with the idea that telomeres are probably controlling the methylation and the glycosylation and things. But the markers for aging, like glycosylation might be a better marker for aging than methylation. I can't really say yet. But I know that telomere length measurement is so difficult to do, and so many people do it incorrectly. There's at least one company that I recommend people go to. I don't know.
You said the company.
Okay, so LifeLink. Yeah, I definitely. If you're going to get your telomere length measured, I think LifeLink is really the only place you should be going.
And not Repeat Diagnostics. Peter Langsdorf's company.
Diagnostics would be okay, except that they don't really measure the percent of telomeres that are short.
Okay, so that's what. Yeah, I mean that is a differentiation between the companies. But my question about the percent short telomeres is when we first did that study with the precursor to life length in Maria Blasco's lab, and we measured the percent short. It was the bottom 3% or something like that. It was, or 3 kilobases and below. They're reporting out 20% percentile. And when we did, remember we did the telomere workshop at A4M, and we had that cohort of people. The 20th percentile had almost like a R squared of 70.7 correlation with the median. So there wasn't, I wasn't getting the same information. It wasn't extra information.
I don't know why they switched to the 20%, or just doing like, I thought it was 2.5 kilobase pairs that they were doing, but there maybe it was 3 kilobase.
Yeah, it was much shorter than, and 20th percentile just is doesn't add that much information. That's my concern about it. I know that the theory, and you've talked about it before, is that it's the critically short telomere that causes the problem. But I don't know that we're getting that measurement. That's my only issue.
I think measuring the critically short telomere gives a better correlation with aging and disease than the average. But I think it's only because the average is so hard to measure. Okay.
Oh, yeah.
And but it's so the percent of critically short telomeres correlates with the average, but it's easier to measure the percent of short telomeres. Now, I'm not exactly certain why repeat diagnostics doesn't report the percent of telomeres that are short, but they're actually looking at whole cells. Okay, so they're looking at the average telomere length per cell as the cell goes through their flow cytometer. Whereas life length has the ability to look at each telomere individually and measure it. But I've noticed lately they're reporting actually average cells too. So I think technologies need to be improved even more. We actually have 2 protocols here that we're trying to develop right now that look at not just percent of short telomeres, but look at each individual telomeres and also identify which chromosome it's on. And we also, essentially doing high throughput sequencing of telomeres, long range sequencing of telomeres, because nobody's really looked at this, but actually one paper right now has come out on this. We don't know that telomeres are all TTA-GGG all the way through. There are probably regions that might be different. Wow. And they might be correlated with disease. Okay, so it'd be good to start finding out. So I think the ultimate would be when we can actually get technique, and we're working on this right now, even though it's going to be unaffordable as a personalized medicine kind of thing. It would be great for research. We're working on trying to make it so that we can sequence every telomere and know what the whole sequence is, what chromosome it came from, what its length is, and then be able to correlate it with all kinds of diseases in very computer intensive using algorithms and stuff like that compared to diseases. But I think that's going to give us a lot of information. Again, funding is the issue. We've been trying to raise funding to do this for quite some time. And even the long range sequencers are more expensive than people want to invest in. I do believe that telomere length measurement can be the best thing in the future. But right now, I'm thinking DNA methylation and glycosylation are probably the best place to go. And believe me, Repeat Diagnostics is a very, very good company. I definitely believe in their company. I just don't. I wish they would provide the percent of short telomeres. I think that information would be very valuable if it's presented correctly, and that's why I'm not certain that it might be a cost thing. It might be in order to make it affordable. Life length has to go with different methods to. Otherwise they won't get any clients to get their telomere length measured.
I think the main reason probably is with them is that they're more focused on the telomere biology disorders and bone marrow transplant stuff, where they're looking just at really short telomeres, people with telomeres in the bottom 1 percentile, under 5, under 4 kb. So the percent shortage doesn't really make a difference. It's, do you have a telomere biology disorder or not? and the mean telomere length that they report out is pretty good for that. They have a good percentiles on that. They're I send lots of samples to them, other doctors do, but I think their focus is on that. I was thinking about asking Geraldine Aubert to come on and talk about it, but I'm not sure they're that interested in it. That's my thinking about it. So I wanted to shift gears just a little bit. We were talking about telomere length and telomerase, but there are these other sort of, and I want to get your take on it, these non-canonical activities of telomerase in the mitochondria. We talk about it shuttling into the mitochondria and the beneficial effects that it has. What's your take on the importance of that role of telomerase? And
I personally, I haven't seen any studies that really convince me that that's for real. It's something that I want to take on when we get more funding. Is I want to look at that more critically, but it's almost like irrelevant. Okay, as long as our cells aren't producing any telomerase to begin with.
Well, that's a very good point there. So, but so I'm just thinking that's a study out of DePino's lab that I think Sahin did,
Yes.
where he showed that there was increased, mitochondrial biogenesis through, the PCG1 alpha and beta, and is that through telomerase tracking into it? No,
that was probably due to changes in gene expression on the main chromosomes that were affected. Because remember, most of the proteins in the mitochondria are encoded by the chromosomes, not the mitochondria DNA.
Right.
And so that was probably an effect of gene expression on the main chromosomes, which was causing the energy levels. But I can't remember the name of the genes right now. The two E. I remember they have a letter E in it. I can't remember the details of those, but those, yeah. But I think that telomere length does have a big effect on mitochondria health. The data that I'm not convinced about is that telomerase, if it does get inside mitochondria, is it actually doing something there? Is it just completely irrelevant?
what would it be doing?
There's no end. Yeah, mitochondria DNA is circular, but it's the question is, does telomerase have other activities besides lengthening telomeres?
Right.
And that I haven't seen any convincing data of. And from an evolutionary perspective, there's a lot of reasons to suspect there's a lot of things that happen in the cell just because why not? It doesn't do anything. There's nothing to cause it to be there, nothing to cause it not to be there. It just is there, and it is completely irrelevant. And so a lot of these things haven't been addressed really, as far as why does it look like telomerase actually gets inside of mitochondria. And I would think there'd be a lot more studies out now if that was actually reproducible by a lot of labs. But I can't say that the data that is published right now is wrong but I'd like to get my hands on that myself and look at that.
And the mechanism through which telomere shortening changes gene expression, is that through that, what's the term that they use? Telomere gene expression change at a distance. I forget what it's called. I'm blanking on it right now.
Cancer sequences or
no? It has to do with the way when the T loop gets smaller, it changes the way in which it touches the DNA, like 10 megabases proximal to the telomere, and so just even a small amount of shortening can change gene expression, and that's why it's. I mean, I've always thought that the theory of it's not only important not to have critically short telomeres, but to have exactly the right telomeres that you start out with. If that mechanism is affecting gene expression, even with a small change in telomere length, then is it important to maintain your telomeres at a certain level, not so much as like you can let them go down to as long as they don't get below a certain length. But we know the aging process starts, and you start seeing aspects of it in the mid-20s, late 20s. So is it that kind of, you talk about gene expression, DNA methylation, sort of picking up the changes in aging, and that's somehow controlled by telomere length. Is it through that way in which the slight change in telomere length changes gene expression?
Well, a lot of what you just said sounded like the theories I've proposed, okay, which haven't been generally accepted by a lot of people. And that's,
I've read some papers. I can't cite them right now, but I have read some papers where they do talk about that and showed some data.
Woody Wright and Jerry Shea did publish some papers showing that they could affect gene expression, and if they shorten and lengthen the telomeres, that gene went up and down, and there was a position effect.
Yeah, a telomere position. That's what it is.
TPE.
TPE.
Okay,
right.
Yeah, I definitely believe in TPE, telomere position effect. But I think of the telomere as like a magic wand, okay? And like I remember when I was in the early 70s, studying molecular biology for the first time in textbooks written by Bruce Alberts, where he talked about, he gave a really good explanation with a lot of papers explaining why genes can be regulated by other elements that are a long ways away, okay? These enhancer sequences that can be 100,000 bases away, but still affect the gene expression of that gene that's 100,000 bases upstream of it. And the DNA folds over, and transcription factors bind to the enhancer and bind to the promoter, and then these things come together and turn the gene on, okay? So I think of the telomere, and the telomere actually does have a lot of similarity to transcription factor sites, okay? And TRF1 and TRF2, which bind telomeres, do have a lot of similarity to transcription factors.
Oh, really?
I think the telomeres are actually working like enhancer sequences, and that they fold over and essentially touch genes, turning them on and turning off just like a magic wand, okay? But the shorter the telomere gets, the less likely it is to reach the genes that are further away. So
And it might change gene expression in some other genes that you don't necessarily is not in the salubrious direction, potentially.
right, indirectly, yeah, those genes, because like one thing that I noticed is that a lot of like zinc finger transcription factors, their genes are located near the telomeres. So the telomeres are folding over, touching these genes, turning them on and off. These are going over, touching other, activating or repressing other genes. Okay, so there is this theory that, you know, I propose a lot. I've got some hypotheses to do testing and things like that. But again, it's you know, when can we do it? How do we do it? Things like that. It's funding and priorities are a big issue. But I do believe that the length of the telomere is affecting gene expression. And the really only paper so far is Woody Wright and Jerry Shea's paper, which was really good, showing that they could lengthen this telomere position effect. The gene was DUX, DUX, I think, the DUX gene, where they showed that they could control its expression, but just by changing the length of the telomeres or its position relative to the telomere. And I believe a lot of genes are regulated like that. We just don't know yet. But you know, people seem to forget about Bruce Albert's textbooks back then, which showed that this phenomena already exists. There's a precedence for it. Why wouldn't telomeres be doing this? Okay, people want to envision some type of chromosomes or chromatin molecules, histones moving along the chromosomes, regulating things, and there's no precedence for that, really, for doing. There is precedence for histones moving along chromosomes, but not long distances that are needed to actually have effect on gene expression. So who knows? But it's all still research. We just got it. But there's what I really like about telomere biology is there's so many good theories that explain everything about everything. Okay, and I say it's the only theory of aging that can do that. Okay, everything else is nothing else. Like you can't say that some of the other things like methylation is affecting telomere length, but you can say telomere length is affecting methylation. Okay, it's and so telomeres. So I compare this to the searching for the source of the Nile a lot. I call it searching for the source of aging. What is the ultimate source of it? Is like methylation a symptom of aging, or is it a cause of aging? Well, I call it both. Okay, I call it a symptom and a cause. But I think telomeres could be the cause of aging, and other things are symptoms and then indirect causes.
So it's the most upstream, you believe, of them. But I mean, actually, just getting back to the methylation, potentially methylation around the telomerase gene could, I mean, affect telomere length. So it could work both ways, potentially.
It's been shown in cancers, yes.
Right. Yeah. Yeah. So I mean, I think that's the whole idea behind a sea change in approach to Alzheimer's disease is in focusing on the glial cells and their caretaker role and their role in keeping the neurons healthy so that they don't start producing a lot of beta amyloid, rather than focusing on removing beta amyloid. And when you talk about curing aging, I mean, I think there's a difference between curing aging, when I think about it, which is someone's old, like an older DePinto mouse, and then fixing them, as opposed to preventing aging, where you keep them in the healthy state and the aging process just doesn't happen. For me, it seems like it would be easier to do that with Alzheimer's than prevent it than to cure it, particularly if you're able to keep those cells doing their job indefinitely. Then, and I think that's I guess where we should start looking earlier on.
But to get a clinical study to use telomerase to prevent Alzheimer's is going to be next to impossible relative to doing a clinical study to actually treat Alzheimer's.
Well, so that's where, the biomarkers, the models, the fMRI and some of the new techniques that they have. I was talking to Tom Dowd about looking at the retinal epithelial cells and how they can be a signal for early changes that could precede Alzheimer's disease, and getting validated biomarkers that, in order to fully validate it, then you'd have to follow those biomarkers for a long time. But it's probably worth taking a flyer on it for potentially, and then it may work in slowing down Alzheimer's and early Alzheimer's as well, potentially. So you might be able to do it that way, and that's where the TAME trial is probably important, because it's going to start to focus on looking at the aging process, and hopefully, when it finally gets underway, COVID put the kibosh on that too. They'll then start to think about things differently. I mean, it's all about the biomarkers. I mean, that's what I've always been involved in, because no one's going to make a career waiting for 25 years to see if you prevent a disease.
And that's one of the reasons why I want to focus on reversing the disease. Because you can see the reversal of the disease a lot faster than you can see the prevention of the disease.
Absolutely true.
But it's like when I was talking about it's hard to get a clinical study for those. I'm talking about getting approval from the FDA to treat somebody with something like a telomerase inducer, because the fears of it causing cancer and stuff like that. Now, some of the other things like the TAME study and things like that with metformin and things, those are different. Those things have already shown. People aren't really saying these are going to cause potential health problems. Okay, so getting a study right there, I think, is a good idea, and I strongly support them and encourage them. And I take my metformin every day, and I've been taking it for 10 years. Because I believe that it's something, but I don't believe it's the end all. I think the end all is going to be lengthening telomeres.
Yeah. So I mean, look, it's been fantastic talking to you. I mean, is there any kind of parting thoughts you'd like to leave? I mean, I think about the future of telomere biology and aging, or just yourself?
I mean, I can't think of anything. There's so much that I could talk about. If we had another hour, I'd love to talk about what is aging, what causes aging, why do we age? My background in evolution gives me a unique perspective on that. But it's a long. There's YouTube videos where I spend a lot of time talking about those kind of things. But it really doesn't help us come up with the cure for aging by looking at those things. And so my focus is on trying to come up with something that's going to kind of cure aging. And again, semantics, my definition of cure aging means preventing Alzheimer's, things like that. So it's just cure aging is just become a coined phrase for my company. And in fact, my father, those are the words my father said to me when I was 10 years old, when he said, "Why don't you grow up and become a doctor and find a cure for aging?" So
I read that on Wikipedia, and I was like, did that really happen, or is that just a great story?
That's true. I'll never forget. I remember where I was, what I was doing. I was obsessed with science and medicine, and I wanted a telescope for Christmas. And my parents got me a really crappy telescope, and I cried about it. They went out and bought me a nice 8-inch reflector telescope from a garage sale. And that night I was out on the front lawn of our house finding craters of the moon, moons of Saturn, rings of Saturn, moons of Jupiter, things like that. And my father just came out and said to me, "Bill, since you're so interested in science and medicine, when you grow up, you should become a doctor and find a cure for aging." And he was serious about this. And then he said, "I don't know why nobody's done this yet." my father wasn't a scientist. He just did not like the whole idea that he was aging. He thought maybe his son, because his son was so obsessed with science and medicine, his son would be the person that's going to come up with a solution. So I got hooked on it. I mean, all through schools. I mean, I used to, even in elementary school, junior high school, everything like that, people would get sick of me talking about curing aging. And high school, college, I did experiments with fruit flies in the early 1970s to try to cure aging. I wish I knew Mike Rose at the time, because, and we should have mentioned Mike Fossil. I just thought, because we were talking about Alzheimer's, Mike Fossil and his company Telomere has got a program underway to try to use telomerase induction to treat Alzheimer's. And I just want to, mention it just to help them get support to get this thing underway. But Mike Rose, I ended up meeting Mike Rose in early 1960s. In fact, Mike Rose and I started a company together called Methuselah Research, MRX. And it was like the next day I learned about telomeres. And so I walked away from the company and brought my father in, and my father took over as the president. Okay. My father and I were like best friends because we were both so obsessed with aging my entire life. This is what we talked about all the time. So when Mike Rose and I and Pravi Sarvor started this company called MRX, and I suddenly wanted to go work on telomeres instead, I brought my father in, and he became the president, worked with Mike Rose and Pravi Sarvor, and made some significant progress. But again, funding was always the issue. Mike's, what the plan was to apply everything, because Mike was the leading, and still is the leading expert in the world on extending lifespan in fruit flies. Even though I'd worked on it before, I did nothing like, in fact, he probably worked on it before I did. I just didn't know he was working on it. But the work I did was nothing compared to the work that Mike Rose did. And when I met and talked with Mike Rose and Perfect, we talked about, let's apply everything Mike Rose had learned in fruit flies. Let's apply that to mice with Pravi Sarvor, who was working on mice colonies. And that was the whole focus of MRX.
But you were going to do that, just breed older mice and see if you could have the same longevity effect. Yeah, it's a very significant effect.
It was a lot of work. It was a lot of work, but it was a lot more work than fruit flies. But all it needed was a bunch of money and a big warehouse where you could have lots of mice and breeding and stuff like that. And then when you got longer living mice, then find out what changed, what genes were selected for. That was the whole plan. And I'm surprised.
Somebody should still do that, right? Exactly.
But the funding,
well, look, I mean, I think there's been a sea change just in the last couple of years in interest in funding aging, and then just recently with Bezos's $270 million, creation of that company, paying the scientists $1 million a year. So I think,
I'm surprised
happen.
Are all over the news raving about the great salaries they have now. Well, that's not the issue. Is curing aging is supposed to be the important things. Now, I'm concerned about the Yamanaka factors. We've been working a lot with the Yamanaka factors, and I collaborate a lot with
Michael West,
Mike, and he and I, we've done a lot of work together on Yamanaka factors, published and things like that. But the Yamanaka factors are really a de-differentiating technique, not a rejuvenation thing.
Right.
And I compare it to, like, does turning a butterfly back into a caterpillar make it younger? Or does turning a frog back into a tadpole, does that make it younger? It just differentiated it. It didn't make it younger. And I'm afraid that that's what's going to happen with the Yamanaka factors. And I'm concerned that these billionaires might have been misled into thinking that this is going to actually be a cure for aging. And maybe it will be. Okay. I mean, it's like, let's get it tested. But I'm just feeling that I hope the investors know that this is really a discovery research, not an actual application to try to get it done. But again, I can't even say that because it might just work. Okay?
What happens to telomere length? Does it get reset with Yamanaka factor?
When the Yamanaka factors get induced, or when you do treat with 4 Yamanaka factors, telomerase gets turned on, and so telomeres get longer. We've done a lot of research here, and we've presented posters and stuff like that, trying different combinations, like 1 Yamanaka factor, 2, all different combinations. So what is it, 4 different factorial, different ways of treating things, and trying to find some combination that turned on telomerase without dedifferentiating.
Right, that would be.
And it never happened. It
Wow.
required, the only time we could get telomerase induced was when all 4 Yamanaka factors were added. And it's like this partial reprogramming, this whole idea of partial reprogramming. I don't know if that's going to really do anything, because it's still going to cause dedifferentiating. It's going to cause telomerase to get induced and make telomeres get longer. But we got to figure out a way to turn on telomerase without dedifferentiating cells. Except we want to dedifferentiate senescent cells into non-senescent cells, and that's the only thing we want to do. And that's been successfully done by Jerry. Again, I mentioned their names a lot: Jerry Shea and Woody Wright. They've done experiments where in vitro they've been able to turn senescent cells into non-senescent cells just by lengthening the telomeres. But it's again, we could talk for hours if you wanted to.
Well, we may have you back for a redux if possible.
Okay.
I think we could talk about aging per se, because I think you do have to really understand why aging occurs to buy into why it would make things a lot better if you can stop aging. Some people are like, well, that's not really going to do it, and that doesn't make that much sense. And so if you really understand the biology behind why aging comes, and there's pretty good, I think, consensus on that in gerontology communities, then I think it makes looking at diseases of aging differently.
So nobody knows what causes aging. Nobody knows why we age. They're all theories. Okay.
Right. Well, yeah, but I mean, there's some consensus on the idea that it comes in through the back door. I mean, there are some people that still think that might be actually programmed. I don't forget which camp you're in, but many people think that it's a trade-off of resources versus the environment, and so why are there species with negligible senescence? Because they don't have a lot of predators. The field mouse is going to get eaten, so there's no reason to invest in keeping his body in good shape. That kind of stuff is pretty well agreed upon, I think. The molecular mechanisms of it are less so.
Everything you're saying is making me hope that you invite me back so we can have a discussion. All right, good.
We'll have that discussion. Yeah, I'll have you back. Very good. Well, it's been a pleasure, Bill, and we'll talk soon. Before we move on to the next speaker, what have you enjoyed most so far? Have you learned something new? Is there a particular speaker that inspired you? Let us know in the comment section below. Hello, I'm Dr. Joseph Raphael, your host of the Telomere Summit. Today, I'm very pleased to be talking to Dr. Gordan Lauc. If you're interested in telomere biology and its clinical applications, I guarantee you'll be fascinated to hear about glycobiology and specifically how IgG glycans can help you take better care of your patients' aging processes. Welcome, Gordan.
Thank you for the invitation. Pleasure to be here.
Yeah, I'm looking forward to talking to you. Dr. Lauc is a professor of biochemistry and molecular biology at the University of Zagreb and director of the National Center of Scientific Excellence in Personalized Healthcare, honorary professor at the University of Edinburgh and the King's College London, and member of the Johns Hopkins Society of Scholars. In 2017, he initiated the launch of the Human Glycome Project and is one of its 2 co-directors. His research team is pioneering high-throughput glycomic analysis and application of glycan biomarkers in the field of precision medicine. By combining glycomic data with extensive genetic, epigenetic, and biochemical and physiological data in a systems biology approach, they are trying to understand the role of glycans in normal physiology and disease. Professor Glycan, we should call you Professor Glycan. Professor Lauc has authored over 200 research articles that are cited over 5,000 times. He was PI and co-PI for 4 NIH and 2 FP6, 7 FP7, and 6 H2020, and 3 ESI funds projects and coordinated 5 of them. In 2007, he founded Genos, a biotech company that is currently the global leader in high throughput glycomics. Welcome again, Gordan. I'd like to start this conversation by having you introduce to our listeners some basic glycobiology, because they may not be familiar with it, as I wasn't until about a year and a half ago.
So glycosylation is the most elaborate and the most abundant post-translational modification. So glycans are chemical structures, complex oligosaccharides, which are being added to proteins post-translationally. So they're not encoded in a single gene, like the polypeptide part. They're encoded in a network of hundreds of genes, which enable proteins like immunoglobulins to acquire novel functions even after the gene for a specific immunoglobulin has already been defined. And I work in the field of glycobiology for, I think, over 30 years now. And we established a field called high throughput glycomics, where we started to analyze thousands and thousands of glycons from different people. So kind of similar to personalized medicine, we are trying to do personalized glycomics. And we already analyzed over 150,000 people. And by analyzing so many people from many really well-characterized cohorts from all around the world, we learned that glycans attached to immunoglobulins are highly correlated with aging. And other people also work on IgG glycosylation. You know, immunoglobulins are the best-selling drugs in the world for a decade already. And we know that these glycans have many regulatory roles on immunoglobulins. And one of these roles is the regulation of low-grade chronic inflammation. So if there is a specific type of glycans, which usually we find in young people on immunoglobulins, these immunoglobulins actually suppress inflammation. They do not allow this low-grade chronic inflammation to be present everywhere. But if glycans are changed, if different glycans are added to immunoglobulins, the pro-inflammatory glycans, which are usually shorter, they don't have a galactose and a sialic acid on the end, they become pro-inflammatory. And this is something what we usually see in old people. We see that people have glycans which are pro-inflammatory, so they're actually promoting low-grade chronic inflammation. And in this way, they are significantly contributing to the inflammaging. So they're doing damage to the tissues, which is then manifested as aging, but also as age-related diseases.
Yeah, so it's a whole new area of biology that's really fascinating. As a biomarker of aging, sort of, you know, telomeres are a biomarker of aging. You have epigenetic DNA methylation. The correlation, is it sort of linear across from, you know, say 20s to 80s, or is there kind of an acceleration at some point? And what's the spread on either side?
So
in terms of years,
it's different in men and women. So when you look at the men have more or less linear increase in glycans from their kind of early 20s, when they are the most suppressing inflammation, to the old age when they're more pro-inflammatory. In women, the curve is a little bit different. So the biggest change happened around menopause. So before menopause, women usually have less of these pro-inflammatory glycans than men. But then in perimenopause, it accelerates rapidly, and they quickly catch up with men and actually go above the men average for a short period of time, and then they kind of look similar later. So it's different men and women, and it does change with time a lot.
And when you give a GlycanAge to they get the test from your lab, and say a woman is 60 years old, is that her GlycanAge against a untreated postmenopausal group, or it's against? So it's against no treatment? Because I mean, that's how you and I sort of met. Because I started ordering your test, and we saw that some of our patients, particularly men and women, but had much younger GlycanAges, and everybody in my practice is on some sort of hormone optimization. There's a big impact there. We were talking about that. I know a lot of our listeners practice hormone optimization, and And so talk a little bit about, that, what happens with hormones, because I know there are other things that impact glycans like lifestyle, body fat, metabolic health, hypertension, et cetera. But I think, one of the biggest impacts is hormones.
So our baseline is the general population. And in the general population, a majority of people are not doing hormone optimization. So I would say the baseline would be people who do not do hormone optimization. And the GlycanAge as index we have is modeled based on these thousands of people we did in the past. So we kind of say your glycans look like an average glycans of a person of 58, 75, or whatever year old. As you mentioned, we got quite puzzled with your data. And actually my statisticians were initially wondering what went wrong and why are all the samples which we are now analyzing coming from your lab so much younger, until we started talking and doing more and more people and realized actually that what you do in your praxis is actually making people look younger on our GlycanAge test. And here I would like to stress one very important aspect, and this is that glycans on immunoglobulins and our GlycanAge, they're not only a biomarker. For example, if you talk about epigenetics, methylation on a specific CpG site is just a biomarker. We don't know anything about its function. And original Steve Horvath's epigenetic clock is something like, I think, 373 CpG marks. Glycans are functional effectors. So these are molecules which either suppress or promote inflammation. So if somebody has more of these glycans which suppress inflammation, we know that inflammation level will be lower. And if there is more of these glycans which promote inflammation, we know that they're actually promoting inflammation. And now we know that this low-grade chronic inflammation is behind so many different chronic diseases that actually we can be very confident that having a lower GlycanAge is something which is good, which is associated with a lower risk of a disease or a milder disease. And something else which is also very important, when we look longitudinally, glycans usually change first. In some diseases, they change up to 10 years before the disease is actually diagnosed. Because, disease, this is a name which was given by physicians for a condition when people appear to the hospital because something does not work. There is pain, some organ doesn't function, and then you get a disease name. This does not happen overnight. This is a long process where the body is first trying to compensate for a problem, and then when all the compensatory mechanisms fail, there is disease. And glycans and immunoglobulins are one of the early things which happen. So before you get ill, your immunoglobulins become pro-inflammatory, they start to do damage, and well, we haven't proven it in humans, we have some evidence in animals that if you improve glycans, disease does not happen. For humans, we only have correlation, glycans change first and then the disease appears.
Which specific diseases are you talking about? Just out of curiosity.
So we have one study where we have shown that by preventing these bad glycans, you can prevent hypertension. And this was done on mice. Mice were fed a high-fat diet, and on high-fat diet, they became fat and they developed hypertension, and their immunoglobulins were hyposialylated. They had less sialic acid, which is this young immunoglobulins. And when mice were fed N-acetylmonosamine, which is a precursor for sialic acid, their immunoglobulin glycans preserved this good glycosylation. So they had young immunoglobulins. Of course, they got fat on a high-fat diet, but they did not develop hypertension. So actually, by preventing aging of immunoglobulin glycans, it's possible to save mice, obese mice, from developing hypertension. For humans, we do not know. We did study a small cohort of healthy people where we did not see a big change after taking manosamine, but we do not know whether actually in obese people taking manosamine would improve glycosylation and contribute to the decrease in hypertension. Something what we published only a couple of days ago is that we were tracking 2,000 twins in UK for 20 years. And then we have shown in the study that, and actually there was a replication cohort in Germany, that glycans today are predictive of hypertension in the future. So maybe this could also work in humans, that if you improve your glycans, you will not develop hypertension in the future.
And well, that's really fascinating. I would imagine that would probably also extend to atherosclerosis. Have you looked at, and there's mouse models of atherosclerosis as well, particularly with the high-fat diet. I wonder whether there's an impact there, because particularly women, you see women going through menopause, they're very well protected. And we thought it was from the estrogen before menopause, from getting atherosclerosis, and then afterwards their coronary calcium scores start going up, their carotid intermedial thickness goes up, and their events start going up to catch up with men relatively rapidly. And I suppose that could be because of the change in the glycans that occurs with the loss of estrogen. Do we have any? Go ahead. Go ahead.
So, of course, hypertension and all cardiovascular or all other cardiometabolic diseases are very complex, and they are very different in different individuals. So we cannot generalize. But I guess that this is one of the important mechanisms, because we know that inflammation is important. We know that these old glycans are pro-inflammatory. And for example, in women, we definitely know that estrogen, actually in both women and men, estrogen is improving the IgG glycan composition. We had one proper randomized placebo-controlled trial where the menopause was chemically induced. And then in a placebo group, we saw a huge increase in GlycanAge. I think average was 9 years. In 6 months, there were some women who aged for 20 plus years in a few months, while in a estrogen group, so people where the estrogen was returned with this estrogen patch, there was no change in the GlycanAge. So definitely estrogen is causative, and it does improve glycans. And interestingly, in men, if you give testosterone, GlycanAge also improves. But if you block aromatase, the enzyme which converts testosterone to estrogen, there is no effect. So it's actually estrogen which is improving IgG glycan in men too.
Yeah, and that's a very, when I heard that, I'd always been cautious with Arimidex or any anastrozole, any aromatase inhibitors in my practice, but other practitioners, I think, use it because I think estrogen is a bad thing in men, but a certain level of estrogen is very important. And here's another reason why you don't want to block it down too much, because you're going to lose that benefit. And the benefit is, just turning to men for a second, the benefit is really quite significant. The average GlycanAge in my practice on men that are on testosterone replacement therapy with good youthful levels is 25 years younger than their chronological age, and that is. It's a fit group for sure. I mean, they're working out, but we see a change when they come in, even if they are fit and with low testosterone levels. So, we're up to about 160, 170 patients now we're looking at. We're going to try to see, what other factors might be involved there. But it's interesting to me that it's even more in men. I don't know if you saw that in your study, whether there was a bigger change in GlycanAge in men when they knocked out the estrogen from the testosterone?
So definitely, this was a different study. And in that study, we did not look at GlycanAge specifically. It was a little bit older study when the GlycanAge we were not able to measure it then. But what we did see is that this change from young to old glycom did happen if the gonadal hormones were blocked, and it did happen if the conversion of testosterone to estrogen was blocked. But if the testosterone was returned and there was no inhibition of this aromatase, then the GlycanAge did not change. Not the GlycanAge, the IgG glycans did not change in the direction of older GlycanAge. So definitely, I think just blocking the conversion of testosterone to estrogen would not be beneficial in respect of inflammation and immunoglobulin glycans affecting inflammation.
Yeah, I mean, I think we know in other things for bone health and for arterial health that it's important to maintain a certain level of estrogen. I always tell my patients, when women come in with postmenopausal women, that their husbands have 3 to 4 times the amount of estrogen circulating in their blood than they do, and that's time to get back up to at least a man's level of estrogen. So I mean, I think that this test that you have available, which by the way is really great because it's a blood spot, can be sent to the patient and readily done, is I think going to transform menopausal and andropausal medicine to a certain degree. I know you're working with Louise Newsom in the UK on looking at, you're doing a trial looking at the effect of estrogen replacement therapy or hormone replacement therapy on glycans. It seems to me that all the benefits that we thought estrogen had prior to the Women's Health Initiative have been turned around with a reanalysis of that data, looking at it with a better lens and the timing hypothesis, et cetera. But having this tool to know whether or not a woman has gotten back her anti-inflammatory glycans, I think could be pretty revolutionary. You're looking at it as a marker for perimenopause and for even potentially adequacy of therapy in menopause.
Yeah. So we haven't published anything on that yet. We only have this study, which was where menopause was chemical induced, but we have some initial data. And something which seems fascinating to me is that GlycanAge can deteriorate rapidly in women in perimenopause, especially in women who had very good GlycanAge before. So we have seen patients changing for 30 years within a year. And interestingly, glycans change before any other symptoms appear. So we had a situation where women were shocked with what happened to their GlycanAge. And they said, but I have no symptoms, I have no problems, everything is okay. And then a couple of months later, all the symptoms of menopause appeared. And this is why, the reason why we see that is that estrogen really directly regulates IgG glycosylation, and it regulates it in a kind of a long-term manner, because IgG we measure today is made in the last couple of weeks. So all the alterations in the hormone levels, which happen at that period, are kind of integrated in a GlycanAge. You can still have spikes of hormones, you can keep other things functioning, but the GlycanAge, immunoglobulin glycans will be changed. And I think this is very similar to way HbA1c, the glycated hemoglobin, can be used to track the average level of glucose in the past weeks. In the same way, GlycanAge can be used to track the level of estrogen in the past weeks. So it could be a very early warning signal that somebody is entering the menopause. And of course, we still have to do a lot of research. We have no ideas whether it's better to start therapy early, whether you get the same result after a couple of months without hormones. There are many questions which we are interested to address, and we are trying to do as properly designed studies as possible. And of course, we are extremely interested to collaborate, especially if somebody already has, hundreds of samples in the fridge. We can do a very quick study to see what happens with a IgG glycosylation in women in this very sensitive period where there are so many changes happening in the body.
So I'm just curious about the change of, if you get a big increase in GlycanAge at the loss of estrogen, I don't know if you've looked at this, but pre-puberty, does a young girl get a change in her IgG glycosylation when she goes into menarche?
So working with kids is a little bit more difficult. So we haven't done so many children, but we have done a couple of hundred. And for example, boys and girls look very similar before puberty.
Right.
And then with the onset of puberty, girls start to change very rapidly. And it's a little bit strange because actually kids, despite, before they develop this hormonal status of an adult, actually have very pro-inflammatory IgG. So young kids look like much older adults in their IgG glycan composition. And we know from experience their kids are actually extremely pro-inflammatory. Their immune response can be very explosive when there is some kind of inflammation going on. And then this is suppressed with the hormones for the reproductive period of their lives. And then they go back as older individuals to be more pro-inflammatory. So I'm actually very enthusiastic about what you do with the hormone optimization. And I think this could actually help alleviate many problems related to inflammation, which is linked to all these diseases, which are now burden for the planet because we're all getting old.
Yeah, I mean, we see with testosterone, for instance, and as well with estrogen, that things like insulin sensitivity improve. You lose body fat, and so all these metabolic changes that occur, there may be some kind of a mechanism through the IgG glycans, which is how this is taking place. So I think it's revalidating this concept that humans, both males and females, need a certain level of estrogen to have an optimal aging, particularly from an inflammatory standpoint. Are there any other genetic factors that figure into this? I mean, we've been talking a lot about hormones. I mean, is there what percent of it is heritable? Do you know?
So we did a number of heritability studies, and for the different glycans in IgG glycan, heritability is between 30 and 75%. So on average, we can say maybe approximately half of the composition is genetic, meaning that if your parents have kind of older glycom all the time, you will be in that direction. Maybe approximately 50% of the variation can be explained by genes, and it's extremely complex network. We have already mapped over 40 genes, wow, which work together to regulate IgG glycosylation, and they're not enzymes which actually put glycans. They're different genes which regulate the immune system. And another very important aspect is lifestyle and environment. Obesity is a very strong factor. So with increase in the body weight, glycans age faster. And we also did on these same 2,000 people, which were tracked 3 times which were analyzed 3 times over 20 years. We have noticed that, and these are mostly women, they are from the Twins UK cohort, which are mostly female. And we noticed that the women which were gaining weight over these time points, they were aging faster than the women who were losing weight or not changing their weight. So by gaining weight, you actually accelerate this inflammatory process. And then by losing weight, you can improve it. So one of the easy things which we all can do is just take care of this extra body fat, which we don't need. And this is also improving this IgG glycan. But interestingly, there does not seem to be a magic diet. We did a study of 1,000 people on 5 different diets, including the high glycemic index one. And each of the diets were beneficial for some people, and they were very bad for other people. So IgG glycan were changing in both directions on all type of diets. So actually, one has to try what works for him or her. And the same goes for exercise. So we all tend to believe that, if you eat too much, you just have to exercise a bit more, and then you're fine. And this maybe works when you're a kid or when you're young, but as you're getting older, you cannot sweat out what you eat. And people who overtrain tend to get also pro-inflammatory and get very bad GlycanAge. For example, initially we were thinking that the gyms would be a good place to start working with a GlycanAge and helping people improve GlycanAge in a gym. But then it turned out that most of the personal trainers and most of the people who are fanatic about going to the gym actually got worse. And if you take middle-aged people and try to work with them intensively, they also get worse. So yes, exercise is important, but there has to be a sufficient relaxation time, and there has to be sufficient time to compensate for all these pro-inflammatory aspects of exercise, because you do need some level of inflammation to start your muscle expanding. But if you do this in a chronic way, then of course you increase this low-grade chronic inflammation, which is bad. So we are now trying also, we are doing several studies to try to find an optimal way of exercising and relaxation, which would help you to improve your glycans. So it's a very interesting field of study too.
What's this? Something's popping up, sorry. Yeah, I mean, I have ultra endurance athletes in my practice, and then even like CrossFitters and people who are really going at it, and one could use it as almost a biological Whoop. You're familiar with the Whoop device that you wear that tells you about the strain on your body? Well, this is sort of a molecular sort of integrator of it over a longer period of time. Like you said, it's 3 to 6 weeks for the glycans to change. And if they're, particularly if they're, pre-menopausal and relatively younger, they're in their 30s or 40s, if their glycans are a lot higher and they don't have to carry a lot of body fat, then they're probably overtraining. And it's a good way, I was thinking it'd be a good way to tell them, maybe you need to put in some more days of rest, and we could track it to see whether your GlycanAge is responding to that. I think that could be another fantastic way, and people who are not even on hormone optimization, to see whether or not. So the opposite problem, people aren't exercising enough and are overweight and are eating too much. You do have the other side of it. I mean, I would love to see people that sort of do Ironman triathlons, and I suppose one guy did one a week for a year around the globe. I mean, what does glycans look like? This kind of stuff is probably not good for you in the long run, and it's good information to tell patients.
This is not surprising because the professional athletes are old in their 30s. And you cannot put too much stress on your body and do not expect to have consequences. And one thing which I forgot to mention, also the psychological stress plays a role. So it's also important to have psychological relaxation, to feel good, and to have sufficient amount of exercise, but not too much, healthy food, but not too much. And then things are in kind of balance, which is, there's no magic. We all know what is good and what is not good. The problem is that we all think that we are fine. Now, you look at yourself in the mirror, you are same as yesterday, you don't see anything, and unless you have some kind of pain, and even if you have, you can take a pill and the pain goes away, and you just go on and go on and go on, but there is a wear and tear in your body which you cannot see. And then usually, at least for men, we stop after the first heart attack or stroke, but then it's often too late. So ideally, I want to get an early warning signal to tell me, you're not going in the right direction. And I don't want to wait for 20 years to get the feedback.
Right.
I want to get feedback as soon as possible. So if I start losing my weight, I want to see an effect in a couple of months. And if it's not working, then I can try something else. And this is where I see a huge potential of this test, which we have developed, is that to help people motivate themselves to do a difficult decision, because skipping that extra cookie is a difficult decision. Skipping that extra beer or whatever is a difficult decision, and people need rewards. And if you get the reward after a few months, you see your GlycanAge is going down, I think it can help people. That's kind of an idea.
Yeah, no question. Do you see any, because I know people practice this, do you see any relationship between intermittent fasting and GlycanAge? or is that going to be correlated to if there's weight loss or not?
So we haven't done a proper study of intermittent fasting. My guess is that it would be beneficial. At least we know that intermittent fasting is good for your microbiome. Then your microbiome, because the problem with eating all the time is that in your guts there is always food, and then the most aggressive bacteria grow. While when your digestive tract is empty, then only bugs which can attach to your gut endothelium and actually graze on the glycocalyx survive. And this is our evolutionary symbiosis between the bacteria we like and our glycocalyx, which is feeding them. And if you do intermittent fasting, then you have these selection periods where you get rid of all the aggressive bacteria and only the ones you like, you keep them. And we know if you improve your microbiome, you will improve your glycans. So we had several studies with a fecal microbiome transplant, and yes, improving the microbiome improves the glycome.
Wow.
So I would guess that the properly executed intermittent fasting after some period would improve the IgG glycome, but we haven't done a study yet. So if you know somebody who might have some samples in the fridge, more than happy to look at it.
I don't know. I mean, Walter Longo might have some. He's done the sort of fast mimicking diet. He's done a lot of research on that. He's a possible person for that. I don't have anybody else that I know that has samples. You mentioned hormones. You mentioned stress, both psychological and physical, as things that affect the IgG glycome. Those are all sort of same sorts of things that affect telomere length and telomerase activation. I'm wondering, and I did a quick look and I didn't see anything much. Is there any research on the relationship between telomere biology and glycan biology?
So we did look in a couple of cohorts. We haven't seen a huge correlation. So I think one of the key problems with the telomeres is that first, how you measure them. Some of the cohorts have telomere measurement which is not extremely reproducible. And the second thing is it's heavily biased with the composition of the cells in the blood. So it's telomeres are a fantastic biomarker for an individual cell, but on an organism level, it's a little bit more complicated. So yes, we were talking to Bill Andrews a couple of times to try to do something together with him. He's also very enthusiastic about it, but we haven't done anything yet. So eventually, yes, we will look whether the same intervention has the beneficial effect for both telomeres and the glycans.
Yeah, we just recently published an effect of TA65 on immunosenescence, CD28 negative cells, and there was a reduction in them, presumably, we think, through telomerase activation, sort of a senolytic-like effect, although we don't know whether the cells went away or whether they were just turned back into CD28 positive cells. But the senescence, the immunosenescence that takes place is, I guess, tracking along, or is there some relationship between that and the glycan change, so that it becomes more inflammatory? I mean, we are now collecting the samples of patients who we have the lymphocyte subsets with the senescent cells, the telomeres, and the GlycanAge, and we'll see if there's something there as our sample size gets larger. But it's not, of course, a controlled trial. But I wonder whether there's any thought about that sort of correlation between the senescence that we know goes up because of replicative senescence, the loss of telomere length, and the change in the glycans, maybe in the. I mean, this is a post-translational thing, so it's not going to be in the B cell, a senescent B cell. It's whatever pathways are affecting that glycosylation of the IgG, and whether or not there's an effect there.
Something what we know, that when you have cells in culture, and when they're getting older, they change glycosylation of immunoglobulins they produce. So definitely there, I would expect to see an effect. But cell culture and the human body, they're two very different things. And we cannot look. We are complicated machines. We have evolved for billions of years to compensate for all type of problems, to fight all possible threats. And I think one of the most, well, we have two very complicated systems. One is, of course, our brain, which is immensely complicated, and the other one is our immune system. Our immune system is keeping us alive against all these crazy bugs which mutate like crazy. They mutate all the time, and we still survive. So this is a very intricate and a complicated mechanism which has to balance fight against infectious agents, against, on the other side, there are tumors which can occur, the mutation cells. So it's, I think this is all very complicated. We cannot simplify it too much. I think, yes, glycans and senescence and telomeres are correlated in a way, but I think they're still a separate aspects of this whole process. So we have to look at telomeres, we have to look at methylation, we have to look at glycans, and then try to integrate all this data.
Yeah, and you get more information. They measure different things, and they're all important processes. And what's interesting is that there are ways to impact them different ways for different systems. And I mean, I think your GlycanAge marker is great because even if you have a good lab like Repeat Diagnostics doing the telomere lengths, and they can do also the neutrophils, which are not subject to changes because they're really reflecting the bone marrow stem cell. It's a longer process. With glycans. It's I thought it was 3 months, but you're saying really 3 to 4 weeks you can get a significant change in the glycans. How often do you think people should test to see what would be effective their program?
Depending what happens. So if there is no major change in the body, it's months. They don't change very rapidly. But for example, now we were doing a lot of studies on COVID in the last year, and people who had severe COVID, we saw a lot of changes in their IgG glycom in a matter of 2 weeks. So they would gain, I don't know, 10, 15, 20 years in a couple of weeks, and it was the worst. So people who died actually had the most significant changes. This is still unpublished, but this is what we have seen so far. So when there is a rapid change in the body, like an infection, and maybe hormones. So what we are doing now is just looking how quickly the glycans would change after the start of the hormone replacement therapy. Then it could be faster. So for example, we have a cohort of women which we are tracking now every 2 weeks to see how rapidly does it change. But in things like weight loss or exercise, usually we don't see anything before 2 months.
And I think you also mentioned when we talked before, or maybe it was in another with Nicolina, that there isn't that much change across the menstrual cycle. So if you have a premenopausal woman, you don't necessarily have to do your test at the same time of the cycle. Is that correct?
For majority of women, there are no huge changes, maybe a few percent. We have seen examples of some women where the changes were more significant. So it would be better to try to kind of match the similar phase of a cycle. But if it's a longer term, it's not so important. But
oh, okay. So maybe try to do the luteal phase or the follicular phase and just keep that same one. It doesn't matter necessarily which one. Speaking of hormones, are there any other hormones that impact glycans that you think significantly, or is there any research on? I mean, I have quite a few patients on growth hormone, IGF-1 increases. I don't know, I haven't seen a signal yet, but
we don't know.
Cortisol, because cortisol is sort of the inflammatory one.
So we haven't seen huge effects of dexamethasone.
Oh, really?
So, yeah, so dexamethasone doesn't do much in a short term. So in the long term, of course, it does. But for example, we know that, TNF is not a hormone, but it's an important signaling mechanism. So if people are taking monoclonal drugs against TNF, we can see a very strong effects in some people, and some people not so strong. So I would guess that many of the immunomodulatory monoclonals could have a strong impact. But these are not things people take regularly. These are
right.
drugs for different conditions. So, the thing is, glycans are very novel. So the technology to measure them in high throughput is here only for maybe a decade. We managed to develop this dried blood stain testing only 2 years ago, because previously we had to have a frozen plasma, and this is logistically complicated. If you collect a sample and then you have to freeze it and ship it frozen, it's complicated. So once we started working with the dried blood stains, everything became much simpler. You can actually ship it anywhere in the world. People can collect sample and mail it back, and it's okay. We can work with the data generated from that sample. So it's still not so many labs doing that. There are over 100 labs in the world which can analyze IgG glycosylation, but not so many of them do it in high throughput. So this is why there is no huge amount of data. It's more and more people working. We even have a thing called the Human Glycome Project, where we are trying to integrate our activities globally, but we still have to learn a lot, because this is a very complicated part of biology and still not so many people working. And interestingly, even at the beginning of this pandemic, the director of NIH was writing a blog about the S-glycoprotein without a single glycan. He just showed the polypeptide part. Well, there is more glycan on the surface than a peptide. And then just after that, people started thinking and saying, wow, there are so many glycans on this virus. What are these glycans doing? And now we know that the glycans are very important, that they're an integral part of not only SARS-CoV-2, but all other viruses. So, there will be more and more glycan work in the future. I'm very positive about that.
And I know you're a very busy man, but I just want to ask one last question with regard to COVID. You brought it up in terms of testing. If a patient has had COVID recently, you would probably expect their GlycanAge to be older, I guess, and maybe take that into account. How long would you wait? 3 months or something?
If the COVID was mild, we don't see much changes. Okay. So asymptomatic COVID, nearly nothing. Mild COVID, nearly nothing. Severe COVID, yes, it can be over a decade, and it can take months for this to come back to the normal level. But it's if it's not, when I say severe, I mean hospitalized in intensive care. So even just hospitalized means moderate, not severe. So for the majority of people, I would not expect to see huge changes.
And how about after the vaccination? Do you have any data on that?
Yes, we have. We looked at some cohorts of people who were vaccinated. There, we don't see any change.
Oh, that's
so
good.
Vaccination does not induce a change.
Good. Well, I mean, I know that you are on vacation. I appreciate your speaking to me, and I know you have to get to your family. As always, it's great talking to you. Any last words you have to say to our listeners?
It was a pleasure chatting with you, and it's a pleasure to work together with you, because we are learning so much from your experience and how you can actually improve people. And yeah, eventually I have to try something myself, but also.
Telling you, come on over. I mean, it's great to work with researchers and sort of put the clinical and the research together to sort of move things along more rapidly. And I'm just very pleased to be working with you and GlycanAge. Thank you very much, Gordan.
Thank you.
If you're enjoying these presentations so far, consider upgrading to VIP on-demand access to the entire summit at a special discount right now. You'll gain anytime access to all of these interviews and bonuses forever, so you can go back and reference this incredible wisdom again and again. Plus, you'll have the added benefit of earning 0.5 CME credits per hour at your own pace, simply by having these informative presentations at your fingertips to learn from anytime, anywhere. Welcome to another episode of the Telomere Summit, sponsored by Dr. Summit. I'm Dr. Joseph Raphael, your host of the Telomere Summit, and I'm very happy to have here today with me Dr. Bill Pawluk, who will be talking to us about PEMF and his interesting journey over the past 30 years toward using it to help patients with many conditions and the aging process overall. Welcome, Dr. Pawluk.
Thank you, Dr. Raphael.
Dr. William Pawluk, MD and MS of Science, is the author of Supercharge Your Health with PEMF Therapy and was recently a holistic doctor in Baltimore, Maryland. Previous academic positions he held at Johns Hopkins and University of Maryland. He has training in acupuncture, homeopathy, hypnosis, energy medicine, nutrition, and body work. He is considered the foremost authority on the practical use of pulsed electromagnetic field therapy in North America. He is interested in holistic pain management, sleep, anxiety, anti-aging, tissue healing, and general health. To promote the healing value of PEMFs, he has well over 50 radio, podcast, magazine, and TV interviews combined. He has also been a co-host of a 2-hour holistic health radio show in Baltimore for 10 years. And most recently, he hosted the Pain Solutions Summit, interviewing more than 30 experts on helping people with chronic pain. He has recently been awarded the ACIM Lifetime Achievement Award in 2019 for work with PEMFs, and is Maryland's top preventative medicine specialist award in 2021. His new book, Supercharge Your Health with PEMFs, is a practical hands-on guide to PEMF treatment, how to select and properly use the right PEMF system for over 80 different health conditions, and cuts through the online confusion about PEMF devices, all based on his extensive knowledge and clinical experience with PEMFs. And we'll show you those books at the end of this broadcast. So welcome, Dr. Pawluk. I'm a bit of a neophyte when it comes to PEMF, but our conversation that we had in Las Vegas was really interesting, and really happy to have you on this episode today. So I'd love to start, as I often do, with telling us about your journey over the past 30 years from sort of traditional medical training into PEMF, and how you got to be where you are in this field.
Well, thank you for that intro. My background, basically, I'm an MD, and I was in charge of a large medical group, 30 clinicians in New Jersey, and we shared patients, did rounds to, mutually on our patients. And a very short period of time, I had a couple patients admitted to the hospital with GI bleeding, and in evaluating that, one of them almost died. And evaluated that, the cause was related to ibuprofen. So both of them had been on long-term ibuprofen. And I realized at the time, when I look at my consultants and I thought about what options were available to me, I didn't see any better options than what already existed. Now, again, that was back in the mid-'80s. So there weren't that many options. So opioids and ibuprofen, Tylenol, and then the consultants and all the things that they did, the procedures, et cetera. So I had to do something different. And it couldn't be within the house of medicine, because obviously I knew what the options were there. And so I decided to study acupuncture. And when I started doing acupuncture, people were refusing to do needles. That was in 1990.
Right. Yeah, sure.
Nobody knew about acupuncture back then, right? So then, having studied that and couldn't use needles, people then thought, well, I have to find a different approach. So I started looking at using magnets on acupuncture points. I knew that in the Orient, in Korea and also in China, to some extent, they used magnets on acupuncture points. Small little tiny magnets, and you place them on acupuncture points and stimulate the point that way. But we know now that you can stimulate acupuncture points in a lot of ways, including light, heat, friction, pressure, and so on. Well, it turns out that the magnets were doing different things than just acupuncture. I would see lesions disappear under the magnet.
Wow.
And I had a personal experience where I had a spider bite on my leg, and I didn't realize I had it, that I'd been bitten. At that point, it was not that itchy or sore or painful. When I looked down, I had a quarter-sized lesion on my leg. So I put a magnet on it, and I was reading on my patio. 3 hours later, gone.
Just a regular magnet.
A regular magnet, fridge magnet.
Wow.
3 hours later, it was gone. I mean, you and I know that a spider bite is going to last a week to 2 weeks.
Absolutely.
I caught it right away. I caught it right at the very beginning. So basically, it just melted away. And that's so that, again, became very intriguing to me. What's going on here? It's not just acupuncture stimulation. There's a lot of other things going on in the tissue related to the magnet. So as I started working with more magnets and discovered a lot of different actions, started using magnets of different kinds around necklaces, bracelets, back magnets, flat magnets, all kinds of magnetic systems. And they were all doing very interesting things. I mean, it was not in my paradigm as an MD, right? To think that what is going on here. In fact, I actually visited with a PhD, chief of bioengineering at Hopkins. I said, what's going on here with these static magnets in biology? She said, I don't know. It doesn't make any sense. And I said, well, it clearly makes sense to me as a clinician because I see the results and benefits all the time. I said, well, I'm used to time-varied magnetic fields, pulsed magnetic fields. I know what they can do, but I don't know what a static magnet can do. So I started working beyond that. That intrigued me as well. So I started working with pulsed magnetic fields, what are called time-varied magnetic fields. So a magnet, a fridge magnet, is a static magnet. It's not varying in time. It's permanent. So if you want to make it more dynamic, you start to vary the magnetic field. Right. And that's where pulsed electromagnetic fields come from. And a magnetic field is produced by a current flowing through a wire. While people are worried about power lines, right? Because of the magnetic fields they broadcast. Right. Even though the current is contained within the power line, it's still producing a pretty large magnetic field. And so I discovered, in fact, that pulsed magnetic fields were much more dynamic, could reach much deeper in the body, could do a lot more stuff, a lot more, provide a lot more help to people with different problems. And along the way, I met a doctor from the Czech Republic, an MD PhD, Dr. Jarabek. And his PhD was on pulsed magnetic field therapy. Oh. Right. So here was an MD, trained MD, and got his PhD in PEMFs. So he translated a lot of Eastern European literature. When I tried to find the literature, the science to support what magnets could be doing in the body, it was very difficult to find, I think, almost impossible. And what I could find was in other languages, foreign languages. So it was in German or, again, the Eastern European languages, Russian most commonly. So he had translated all these studies basically for his PhD. In addition, he was recruited by the government of the Czech Republic to be a consultant for them to help them advise about approving PEMF therapies in the Czech Republic, primarily for rehab purposes, because that's what he was doing, mostly rehabilitation medicine. And in the end, he provided me his translated review.
Oh, great.
And so we worked together and produced a book called Power Tool, Magnetic Field Therapy in Eastern Europe, How Magnetic Fields basically work. And there was the research evidence over 30 years of science in Eastern Europe. Okay, wow. I had the science. Right. For the static magnets and the pulsed magnetic fields. And that book then basically provided a really solid foundation for me to understand what magnetic fields were doing. I expanded my work with it, and then bringing us up to date now, I published the book with him, and I'll show you that in a second. Now it's a fairly technical book.
Step back a little bit. There you go. Okay.
Right. So it's still available. You can get it through our office here. I did sell it for a while on Amazon. Mm-hmm. So anyway, this book has lots of citations and lots of evidence and talks about different uses and different clinical specialties. Well, then I produced a website, put as much content on as I could, acted as basically a consultant or an educational resource about magnetic field therapies. And subsequent to that, then I continued to review the science myself, whatever I could find, in addition to what I had with Dr. Jarabek. Also got access to an abstract library of over 30,000 abstracts on the effects of EMFs and PEMFs on the body, or on biology in general. So, okay, now we have some pretty solid science. But, I was one person. I had all this knowledge and nobody else did, other than the experience people had in Europe. So I kept working with magnetic fields and then produced the website, drpawluk.com, and there's a lot of content on there. But the content on the website is limited. Websites are limited in what you can say. It's almost like sound bites. You have a small amount of information you could provide. So eventually then I produced a book called Power Tools for Health, How Pulse Magnetic Fields Help You. In that book, then I have about 500 references. Part of the problem clinicians face is that there is a journal called Bioelectromagnetics, Journal of Bioelectromagnetics, But the problem with that journal is that it is mostly basic science, very little clinical science in it. So that it doesn't help, the clinician basically to figure out what to do with magnetic field therapy. And so we published the book, Power Tools for Health, provided 500 references to back up all the actions I discussed. And in my work, my research, I discovered that magnetic fields basically have about 25 to 27 different physiologic actions in the body.
Yeah, let's talk about some of those. And then also, if you will, I know some people are thinking, I was thinking about it, that there's electromagnetic fields and there's, we talk about EMF being not good, being bathed in EMF, your cell phone, your router, et cetera. But then there's also EMFs that are beneficial, pulsed EMFs. And it's, my understanding is it's about the frequency and the intensity. So if you can get in a little bit about that so that the listener's not confused about, well, I'm supposed to avoid them, but on the other hand, they can be healing. What's going on here?
Unfortunately, the terminology is common between the two. So
Right.
PEMF stands for pulsed electromagnetic fields.
Right.
And EMFs are electromagnetic fields without distinction. Right. So unfortunately, it shouldn't be pulsed. It shouldn't be EMFs. It probably should be. In fact, I even consider EMFs environmental magnetic fields.
Okay.
As opposed to pulse magnetic fields. The basic distinction between them is that EMFs are essentially broadcast into space. They're non-directional. They can be somewhat directional, but basically they're broadcast into space. So that's considered an open loop system. Magnetic field just goes out into space.
Right.
Current producing a magnetic field is contained, it's closed. My thumb is the current. This is called the right-hand rule. So the direction of the current flowing produces a perpendicular magnetic field to the direction of the current flow.
Okay.
And with every pulse of the current, what you get is a magnetic field that opens and collapses. In other words, it goes back to zero. It goes from zero out and then back to zero. It's not broadcast. It's contained.
How far does it go? And it just depends on the intensity.
Depends on the intensity primarily. Intensity and frequency to some extent. So very extremely low frequency magnetic fields, let's say under 100 megahertz, I would say under a kilohertz, have basically can go quite a long distance, but it's not a wavelength. It's a pulse.
Right.
That's why they're called PEMFs, pulsed magnetic fields. So because they're pulsed magnetic fields, they're designed for therapeutic purposes. They could be designed for other purposes as well, like in engineering and equipment of different kinds, including in the medical environment.
And I think the lower frequency, I guess, in intensity differs from the higher intensity broadcast EMF in that those EMFs, the latter, can heat up tissue and cause damage and change gene function versus the lower.
That's the key.
Right.
That's the key. So most of the frequencies that we're concerned about, most of the EMFs we're concerned about are microwave frequencies.
Right.
They're extremely short frequencies, like our microwave ovens, right? We use that principle to heat food.
Right.
Right? Because it absorbs the frequency. So these frequencies are absorbed by the body. They don't pass all the way through. Whereas the pulse magnetic fields pass completely through the body. The body is completely transparent to a pulsed magnetic field. And that's valuable because what it's doing then is it's not staying resident in the body. It's not used up by the body. It doesn't matter whether you have clothing on or not. It doesn't matter whether it's muscle or bone or brain or a skull. It's going to go all the way through. That's just the general principle about EMFs. And that's useful for us. EMFs are designed for other purposes. They're designed for communication.
Right.
Right? They're not designed for therapy. So they may have incidental effects on the body, as you mentioned, DNA damage and heating and so on. That's where the risk comes in. So for example, if you hold a cell phone to your ear, almost doesn't matter how long, but the longer, the more effect you're going to have. You'll see that the ear turns bright red. In most people. So that ear is effectively getting cooked.
Even if you're not touching, because I mean, obviously touching it would be.
Doesn't matter. Yeah, you can hold it like 2 or 3 inches away from your head. But because it's close enough and the intensity is high enough, then that dose, that exposure dose, is going to create physiologic actions. Now, EMFs in general most people are not exposed to EMFs in high doses, except unless you're working on towers, cell towers. You're actually somebody working on a tower, then you're going to be exposed very close to that field. Now, all magnetic fields, like any other radiation, whether it's sunlight or sound or heat or cold, drops off extremely rapidly according to the inverse square law. There are other laws that can be invoked as well, but I use the inverse square law. And we use the inverse square law for radiation dosing, right? That's a classical use for inverse square law. So all of these fields drop off very rapidly. And so you have to calculate the dose based on the distance that you're targeting. And PEMFs are the same. You should calculate the dose based on what you're targeting and what your intentions are. So PEMFs basically are very, very, very safe.
And much lower frequency and much lower intensity as well.
The lower frequencies. If you're under 100 megahertz, usually the heating effects are minimal.
Right.
And most of the heating, in quotes, heating caused by PEMFs is actually due to increases in circulation locally. So you get local heat because you've improved circulation.
So we're all familiar with MRIs. Where do they fit in that range?
Well, MRIs there's two aspects to MRI: radio frequency fields and a huge static magnetic field. So the radio frequency fields can have a potential for harm, and there is some discussion about MRI having harmful effects. But you and I know hundreds of millions of people have gotten MRIs, many MRIs with virtually no damage, and the people who work in MRI environments are not reporting significant health issues.
Right. Okay.
So they're not therapeutic. High frequency and static field. So there was a study done at McLean Hospital. They had reports of people who had their depression get better after an MRI.
Huh.
So based on that evidence, they conducted some research to look at the use of magnetic fields for the brain for MRI. And of course, now we know from PEMFs, and now we know that there are devices approved called RTMS or TMS, transcranial magnetic stimulation,
right,
for treatment-resistant depression.
Right.
And there are also fields approved called TTL, basically for glioblastomas. And they're approved by the FDA for that purpose as well. So there are uses of those other kinds of fields to some extent. And there are magnetic field therapy devices that are approved by the FDA for healing non-unions. Now we're using RTMS for depression, which is still a pulse magnetic field, high intensity pulse magnetic field. And that's approved for depression. There's also a field approved for pain and wound healing.
Okay, so I think we have a good idea about what the range of electromagnetic fields are, from damaging to therapeutic and in between, diagnostic. Can you tell us, I'm really curious to know, what is the mechanism for the therapeutic effect of these lower intensity, lower frequency? I know there's something to do with the mitochondria and ATP, membrane ion channels. Let's talk about that.
Right. So the primary law of physics that governs the effects of magnetic fields on biology, particularly, is Faraday's law. And Faraday's law says that as you increase your charge, as the charge increases, so the magnetic field increases from 0, say, to 100, then that happens over time. So Faraday's law says dB, the change in B, the intensity, divided by the dt, change in time, gives you something called Tesla per second. That's the measure of the likely induced field. So the higher the dB dt, the greater the induction of charge in the body. So a pulse magnetic field passing through basically ions, electrical charges of any kind, ionic charges of any kinds, will create charge, will create more energy. In other words, will create more electrical charge in the tissue. So the dB dt of a magnetic field passing through the body is where the action is. It's the induced charge in the body. Now, if you apply an electrical charge to the body, that's called capacitively coupled electrical stimulation. And that's limited by the body's ability to absorb that electrical stimulation. Let's call that controlled electrocution.
Okay.
Right? It doesn't go deep into the body.
Right.
It's very superficial. whereas magnetic fields are inductively coupled electrical stimulation. So it's inducing charge in the body.
Right.
It's coupling to the charges in the tissues that are there already, amplifying them significantly, and that extra charge then is used by the body to produce all the physiologic actions of magnetic fields. So those actions in my book, the Power Tools book, I list 25, and since then I've discovered a couple more. So some of those actions include acupuncture. So that's how I got started on this journey in the first place, trying to find a solution for pain that was not conventional. And acupuncture certainly does that. So magnetic fields clearly stimulate acupuncture points and meridians.
Okay.
And if you stimulate the whole body with a magnetic field, use a whole body magnetic field pad, you're basically stimulating all the acupuncture points and meridians in the body simultaneously. So rather than having to be directed, and ancient science, ancient acupuncture, that's the tool they had. So there was at one point at a time or multiple points at the same time. Well, fortunately with magnetic field therapy, because now we've actually discovered, science has discovered that the acupuncture meridian system is a DC current system.
Oh, really? I did not know that.
From a engineering perspective, it's called a lossless. There's no loss in that system. And each acupuncture point is basically a capacitor. So it takes charge from the previous acupuncture point, builds the charge, and sends it on to the next one. That's what happens in a DC current system. So it's a DC current system, and because it's a DC current system, it applies to Maxwell's equations and Faraday's law. So then basically you can amp up that system using magnetic field therapy. And I measured that. Actually, we did some EMG studies looking at magnets and the acupuncture points, and we certainly found effects. But there's plenty of science about that. In fact, I teach a workshop to acupuncturists about why magnetic fields work on acupuncture and how to combine acupuncture and magnetic fields to get even better benefits.
Okay, so there is, we know that acupuncture works, and we've all had patients have beneficial effects on it, but there's now good science and physics around exactly why it works and how it works. But you're saying like sort of an electromagnetic field mat is almost better in some ways for general therapy.
For general therapy. So one of the actions, physiologic actions of the magnetic fields then is the acupuncture stimulation. It's stimulating the meridian system, and then you get all the derivative benefits of stimulating an acupuncture system.
Okay.
Yeah. So that's one of the key elements. That was one of the first things I discovered in my own journey. Since then, we've also discovered that magnetic fields have antibacterial, basically microbiological benefits. They have anticoagulant effects, naturally, almost equivalent to aspirin itself, much safer than aspirin. They have anti-edema effects. They have anti-muscle relaxation effects. They have increased autophagy. They improve autophagy, not in a bad way, but they improve autophagy.
And how do they do that? Just wonder if that's been worked out.
Probably because of the charge, partly because of all the different effects that it has on the cell membrane. And so we talked about, you mentioned mitochondrial function. So mitochondrial function is improved. And we know research shows that magnetic fields increase ATP production by between 100 to 600%.
What? That's a lot. I'm surprised the Tour de France guys aren't using it, or any. I mean, are a lot of endurance athletes tapping into it?
There are more and more and more. It's hard to get through the trainers. It's hard to get through the orthopedists to get to the athletes. But more and more athletes are discovering these benefits. In fact, anecdotally, I had a guy who was number 3 in the world with golf ball driving.
Really?
Long distance driving. How far can you hit the ball?
Yeah, sure.
Sure. He was number 3 in the world into his 40s. And he was using magnetic field therapy every single day. He would use it before competition, and he would use it right after competition. Anecdotally, a friend of mine, an MD, worked with American Olympic teams. And they would do tryouts, they do competition and so on. And he said, our guys, in this case it was guys, our guys would take 2 to 3 days to recover from prelims or competition. The Russians and the Eastern Europeans were back there next day like robots.
Well, yeah.
Bang, back up. And he couldn't figure out why. What's going on here that they are doing that's different than what we're doing? He walked past their camp they're laying in magnetic tubes.
Ah.
And that's not doping.
No, it's not. It probably will be at some point. Maybe, I don't know for sure. I mean, that's an interesting thing that WADA would have to deal with to decide whether or not. I mean, I would hope that they wouldn't make it because it's helping people to recover. But
it's physiologic. It's
yeah,
not pharmacologic, right?
Right.
And it doesn't matter whether you use infrared or whether you use sound. There's so many ways of stimulating the body physiologically. EMFs or PEMFs are actually just another mechanism. It's a physical therapy mechanism, basically.
So we're talking about recovery in athletics here. There is, and I want to get back to that in a way in a second, but if you had to think about what the sort of the number one treatment indication is for it, tell us what that is. I mean, I'm kind of assuming it's pain or the non-union kind of stuff, and then we'll get into sort of how to use it for preventative and anti-aging purposes. But let's talk about that first.
So I'm going to preempt you if you don't mind, a little bit. Let's go back to the mechanisms.
Okay.
Because when you understand all the mechanisms, then you can say, oh my goodness, what else can we do with this stuff?
Right. Okay.
Right. So we talked about edema, talked about muscle relaxation, autophagy, ATP, circadian rhythm rebalancing, stimulating collagen, hyaluronic acid, GAGs, detoxification, brainwave tuning.
I want to talk about that for sure. Yeah, absolutely want to talk about that.
Right? Like gamma waves, stimulates growth factors, nitric oxide, tissue regeneration, stimulates stem cells, balances immune function, stimulates nerves and nerve conductivity, both up and down. Oxygenation, improves oxygenation significantly. Transmembrane oxygen transfer in the lungs, and then the release of the oxygen in the tissues enhanced by PEMFs.
And how does that happen? I'm curious about how that happens physiologically.
Well, first of all, if the cell membrane is, we open membrane channels, ATPase, sodium, potassium ATPase, calcium, magnesium movement across cell membranes. In injured cells, that stops. It doesn't work. It's dysfunctional. So magnetic field therapy seems to open the membranes and allows those processes to reactivate. So it's basically rebalancing the internal cellular environment with the external cellular environment, and you get better electromagnetic movement in the tissues. That then allows oxygen transfer. So PEMFs affect oxygen carrying capacity of hemoglobin.
Oh, really? Just like being at altitude, sort of?
To some extent, yes. Whatever the altitude happens to be.
Mm-hmm.
So basically, when you're then transferring oxygen from the lung, from the alveoli, past the membrane, if you're opening these channels, you're then increasing the ability of the oxygen to transfer. And then because of the effects that it has on the red cell membrane itself, then you get better oxygenation of the red cell. And then once the red cells get into the capillaries, into the smaller spaces, they release the oxygen better. And once it's released better, then obviously you have more energy for the cells as well. So oxygenation is one of the key principles. I actually did a pulse ox study in an acupuncture practice, looking at pain patients in an acupuncture practice, and doing pulse oxes before and after magnetic field therapy. And we had a statistically significant increase in PO2.
Wow.
Now, you're not talking about going from 67% SaO2 up to 97%. We're talking about, again, 1 or 2%. 1 or 2% is still pretty significant.
Interesting. Yeah. So we understand the mechanism. It's used clinically for which?
Okay, so if you think all the different mechanisms, it really doesn't matter what you use it for. All disease has most of these mechanisms in common, and probably the most common in common, the final common pathway is probably cell injury.
Mm-hmm.
An injured cell has a series of events that happen in that cell, and that cell then has to adapt to that imbalance, to the dysfunctional change, and that dysfunctional change slips down that slope of dysfunction until finally it reaches a point of irreversible injury. So if you're targeting the cell, let's say on a regular basis, cells on a regular basis, you're able to restore those functions: membrane stabilization, ATP production, genetic stability, providing more nutrition to the cell because of the improved membrane transport, getting rid of detoxification, getting rid of waste out of the cells better. So all of these things begin to happen. So it doesn't really matter what the condition is. We put a label on the condition, but what are the physiological? So when I look at a problem, somebody says, I got this problem. I had never heard of it. So I go look it up. What is the pathophysiology of the problem? If there's any of these mechanisms in play in that problem, then magnetic field therapy is going to help to at least some extent.
Mm-hmm.
But what drives people to seek magnetic field therapy?
Not
Cost.
Oh, okay.
Well, pain, I mean, it's the discomfort.
Right.
So pain is the biggest driver. People are going to spend more money to deal with pain than almost anything else.
Right.
And it's the most common. Since you have 100 million people in the US with chronic pain, that's probably one of the most common problems that we deal with.
For sure. Yeah.
Right. So pain is probably the most commonly used. That doesn't mean it's the best, or that doesn't mean that it should be the only one. But again, if we understand mechanism, then we can understand application.
So for diabetic neuropathy, does it help with even things like degenerative disc disease and spinal stenosis and that sort of thing?
Yeah. So again, going back to mechanism. So with spinal stenosis, the cause is either going to be a disc, usually disc, or arthritis.
Right.
Now, you're not changing the arthritis and you're not changing the disc. Those are mechanical.
Right.
And magnetic field therapy doesn't fix a mechanical problem. If your arm is hanging off your shoulder by a thread, then you got to sew it back.
That's right.
Right? So what we do then is we're decreasing mechanisms again, the inflammation, irritability of the nerves, the tissues, then they cause problems with function. So if you can reduce the swelling, and plus magnetic field therapy has a natural anti-nociceptive effect.
Oh, okay.
That's been proven. So pain reduction by itself without any other mechanism also happens with PMF. So one of the first things I tend to see with people with pain is the pain goes away. But I know that's not a solution.
Right.
It's making the pain better. But what we want to do is to deal with the cause. We want to be able to reverse the problem if we can. And so with the back pain, spinal stenosis, if you reduce the edema, you reduce the irritability of the nerve. So you anti-nociception, you reduce the edema, you reduce the pressure on the nerve from the edema. And then hopefully, if you continue to do the therapy over time, you may be able to stop the progression of the arthritic process. And in terms of discs, you may be able to hydrate the disc better because the disc is hydrated passively.
Uh-huh.
Right? There's no the blood vessels going into a disc.
Right, sure.
So it's by osmosis. But if you increase the blood supply around a disc, because often with a disc problem, you also have muscle spasm, reflex muscle spasm. So that tends to aggravate the problem, compression, and makes it worse, right?
Sure.
So relax the muscles, increase oxygenation, increase hydration, and then maybe longer term, you're going to get a stability of that disc problem. So you're working at multiple levels, basically, with the magnetic field therapy.
And would that be for sort of the back issue? And now I'm thinking about myself because I got a little spinal stenosis. Would that be lying on a pad then, or would it be more directed?
It depends on what you have and what you buy. So I happen to be, my philosophy is if you're going to get a magnetic system anyway, if you're going to make that an investment and you get a proper magnetic system, you should be doing health maintenance anyway.
Right.
Right?
Sure.
You're already good because the biggest investment is the control unit. The applicators are less expensive. So get a good control unit and then add whatever applicators you want. So yes, whether it's whole body or local. In the case of, if somebody wants to limit how much they spend, they say, fine, then you need a high intensity local applicator. And you could treat locally. And you're going to get better results faster, typically with higher intensity. And we can get into that in a second with inflammation. Because PEMF stimulate, this is one of the new mechanisms I found since I wrote the book, it's the adenosine receptor. The magnetic field therapy addresses the adenosine receptor. And you need about 15 Gauss optimally at the adenosine receptor to decrease inflammation. Now, the studies were done on adenosine receptors on neutrophils. So where's inflammation?
Right. Yeah, it's coming from neutrophils. Yeah, sure.
everywhere, anywhere in the body.
Right.
So if you get the right magnetic field intensity to address the neutrophil wherever you have inflammation in the body, you're decreasing the inflammation through, at least through the adenosine receptor mechanism.
Well, it sounds like virtually any time you have trauma or wounding, and that would be, from an accident or from a diabetic ulcer or something like that, or even post-surgically, PEMF should be used.
Absolutely. I'll give
How
you
come it
another,
isn't?
I'll give you another anecdote.
Yeah.
Case study. So myself, I had an elective appendectomy. I had a small fatty tumor at the entrance to my appendix during my routine colonoscopies. And so we decided that we had to find out what it was. They couldn't just snip it out. You had to remove the appendix. So I had an appendectomy. It was elective. Had it done on Friday at midday. I was back to work on Monday.
Laparoscopically it was done, but you had
laparoscopically.
Quick healing because you were
using magnets on my stab wounds.
Right. Ah, okay. So then that also then begs the question, you mentioned collagen, gags, aesthetic use of it, like for facial rejuvenation or wrinkles?
Well, you can. I mean, theoretically, you may have to, you're paying a lot of money for a piece of equipment.
You pay a lot for Botox too, so I think you could.
Well, this is true, and it doesn't last.
Right, and
right?
Fillers. So I'm curious about to what extent you have collagen increase, and the glycosaminoglycans increases. I mean, are there studies that look at
yes,
PEMF devices?
Actually, in the Power Tools for Health book, there is actually a section.
Mm-hmm.
Let's find my spot.
There you go, right there. Good.
So there's actually a section in there on collagen and gags and hyaluronic acid. So you can get somewhere between 25 to 30% increase in collagen production.
That's a lot. That's a lot. And over what time period?
Well, again, the treatments typically, in most research studies, the treatments are like 15 minutes to half an hour, and then you repeat periodically. So probably over 2 to 3 weeks.
Really? That's pretty impressive. Okay, well I mean, I know we have people that practice aesthetic medicine listening. And so I wonder, whether that's an area that they could enhance their practices as well with one of those devices. It's, I think, certainly more physiologic and less invasive than, other things. Hopefully, if it works, 25% is a pretty big increase in collagen. So that would be interesting to see. People that are using it in aesthetic practices?
Yes, there's there are biases. We all have biases, right?
Sure.
So it depends on who's been sold what by whom.
Right.
Right. In the aesthetics world, they tend to be a lot more high frequency oriented.
Right. Yeah, sure. The lasers, et cetera.
Exactly. And even light therapies. So the more mobile and useful it is, the faster it gets the results. So some technology will produce results better and faster for certain problems than others.
Mm-hmm.
So the answer is yes, those doctors who are willing to do this. But I find that magnetic field therapy is much better. The kind that I recommend and use is much better in orthopedic applications.
Oh, okay.
And post-operative applications than it is in dermatologic.
Okay.
Because you need very rapid results, right? Short treatment times, very rapid results.
But if you're a home user, you have the time and the luxury to do it on yourself at home.
And if you're doing whole body therapy anyway,
right,
you're getting that benefit anyway.
Oh, okay. Point well taken. Good. So you mentioned TMS and I know I read on your website this idea of entraining brain waves. You get the same frequency in the EMF, the pulsed EMF, and you can get potentially a beta for higher concentration. Can you pulse it at an alpha for the same calm inducing that you get with meditation or neurofeedback?
Absolutely. There are plenty of devices being marketed now that do that kind of work. And I did audiovisual entrainment years ago, decades ago. And that's how I got these concepts. And then I've had some additional training in the past 3 or 4 years in neuroscience using basically entrainment processes for, it's called, neurofeedback, essentially.
Right, yeah.
So with the brain, frequencies now become more important. Most of the time, my emphasis has been on intensity. particularly if we think about the adenosine receptor, the optimal magnetic field intensity is about 15 Gauss. Then you have to, calculate the magnetic dose that you need at the tissue that you're targeting for inflammation. So for the kidneys or the lungs or the heart or a lower back. So for these kinds of purposes, for entrainment purposes, you don't necessarily need as strong a magnetic field. But we've designed a device called the Flex Pulse. So I was involved in the design of that with a company in Germany. And so we have individual frequencies. So we'll have delta, which I use for sleep. And when I'm flying across the country, I fly delta. I do a magnetic therapy machine across my head or the base of my spine over the RAS. And I'm okay.
Very good. So you can go ahead.
So then you design frequencies for delta. You can do theta for meditation. You can do alpha for relaxation. You can use beta for alertness. A case in point. I had to pick up my daughter at the airport in Baltimore about 2 o'clock in the morning. I normally go to bed at 10, and I knew I was going to have a problem. If I drank coffee, I'd be up all night. So I did magnetic entrainment using beta over the RAS, small disc over the back of the head, and I was wide awake. Got to the airport, picked my daughter up. I didn't have hangover, I didn't have a buzz. I was just alert. Got home, took it off, went right to sleep.
Nice. That's very interesting.
It's increasing the amount of frequencies in the brain that are now oscillating at the beta level. That doesn't mean the whole brain's oscillating at beta, but enough energy, enough oscillation is happening that now consciousness shifts into that level of awareness. So you can tune the brain to those specific frequencies, and the Flex Pulse does that.
It's called the Flex Pulse, and
Flex Pulse,
it's a portable device to be used.
Portable, battery operated. Yeah.
Wow, very, very interesting. So we've been having a really good conversation, and anything in terms of healing is obviously of interest to our listeners. But you also did mention that there are a few studies looking at the effect of PEMF on telomeres or telomerase, and also I would think then through that on senescent cells, which could potentially help with many conditions of aging. Can you talk a little bit about that?
So I could only find 2 studies that specifically looked at telomerase. And one of them was done in leukemia cells. And what they did is they looked at the use of DMSO to stimulate the cells, the red cells, and found that the DMSO caused a 100-fold increase in telomerase activity versus sham. DMSO caused a 100-fold increase in telomerase activity.
Well, that's pretty wild.
On red cells, obviously in culture.
Right.
Okay. Now, what happens in the body,
Yeah, sure.
And of course, the dosing of the DMSO and so on, which you could do more in vitro than you can in vivo. What they found is that the PEMF activity, and I have to read here, exposure to 1,000 to a 10 Gauss magnetic field reduced that increase in telomerase activity. I'm sorry, it was a decrease, 100-fold decrease in telomerase.
Right.
Is not what we want to see, right?
Well, in a cancer cell, yes, it is what you want to see, but not in a healthy cell. So interesting.
So in a leukemia cell model, they found a 100-fold decrease in telomerase in versus sham exposed. Now, with magnetic field therapy, 10 Gauss, that field reduced the decrease in telomerase by 10-fold, from 100-fold to 10-fold. So in a sense, it has a protective effect against the supposed negative effects on telomerase in the leukemia model.
Yeah, I mean, you want the opposite to happen in cancer, and telomerase is constitutively activated in cancer cells. I mean, I would think that just from the stress reduction and other beneficial effects, reduction in pain, that over time you would see either a slowing of telomere attrition, just as stress reduction, meditation, all those things help. Those studies just probably haven't been done, but that would be interesting to see. So from a standpoint of practical,
there was another study too, and X-ray exposed ovarian cells. So these ovarian cells were exposed to 10 gray units of X-rays, and they increased telomerase activity by 2.6-fold. So X-rays increase telomerase activity by 2.6-fold.
Okay.
All right, and then so that's a negative action, and we know that, right? X-rays cause radiation damage.
Right. So that's interesting. Go ahead.
All right, so then the magnetic fields were applied as a control, and they marginally decreased the telomerase. They barely touched the telomerase production initiated by X-ray. So that, I wish it would be more, but again, in this case, it's not a negative effect.
Mm-hmm.
In other words, it didn't decrease the telomerase activities to a point where the radiation would be even worse.
Right. Yeah, I mean, I'd be curious to take a closer look at those studies. So they're on they're on the website or in.
No, they're not.
Oh, okay, well, I'll take a look for them. Maybe you can
These
send
are
them
in
to me.
unusual places. I'll have to show you. I'll have to send you the abstracts directly.
That'd be great. That'd be great. So from a practical standpoint, in terms of implementing PEMF in a longevity practice or an age management practice or in your own personal life, what kinds of recommendations is you're talking about daily exposure to PEMF, and then of course localized for particular treatments, higher intensity, et cetera. But it sounds like that's what you're recommending, and then perhaps the flex pulse for sort of specifically brain and mood stuff.
All right, but if we think about the things that tend to shorten telomeres.
Okay. Yeah.
Right.
I mean, all bad habits shorten telomeres.
There you go. So stress, obesity, basically
sedentary lifestyle. Yeah.
Sedentary lifestyle. PEMFs increase stem cell production by about 65%.
Wow, that's a lot.
Between 35 to 65%. And most stem cell lines, so we're talking about mesenchymal stem cells, which are the most common stem cells, right? Then their production is increased and their differentiation is improved. So PEMF therapy acts on basically the entire stem cell process. They increase production, they improve differentiation, they increase survival, right? And they increase the longevity of the cells that were produced. So PEMFs act on the whole stem cell process from beginning to end. And I'll give you an example, a clinical example. I had a 3-year-old child. Now, 3-year-olds have a lot more stem cells than 70-year-olds.
Right.
Right?
Yep.
Huge difference. So this little girl, cut off the end of her thumb in a door jamb. Avulsed it.
Oof.
And the father contacted me right after that happened. He had a small portable magnetic therapy device that he purchased from us. He said, "What do I do with this?" I said, "Well, my recommendation is that have the surgeon reattach it."
That's good advice.
Right? Have the surgeon reattach it because, if it doesn't work, you're back to where you were anyway.
Right, sure.
Right? So they reattached it. And I have pictures in my book where you can see the suture marks. Clearly it's black because the blood supply was disrupted completely. So she did an hour and a half to 3 hours a day of this magnetic field directly on the thumb. 12 weeks later, she's regrowing her nail.
Wow. And so it was the nail that came off, not the whole
thumb.
The whole thumb.
The whole thumb.
So
past the joint.
And so the nail growth, and she had full function and everything.
Yes.
Wow. Yeah. Well, there's some processes being induced that's similar to, sort of the tadpole that can grow the tail back kind of thing where, and that likely involves telomerase activity in stem cells.
Stem cells, telomerase, stem cells and telomerase go together, right?
Yeah, of course. Mm-hmm.
Most stem cell activation involves telomerase.
Absolutely. That's why stem cells are different. They have more telomerase activity and continue to replenish degenerated tissues with it. Telomerase is turned down as a supposed tumor suppressor mechanism in younger people so they don't succumb from cancer. But later in life, that is a deleterious effect of it because we're living longer. We need that regenerative capacity. So that does kind of pull it all together there. I think, what's your thoughts about the future of PEMF and how it's growing? I mean, you're obviously doing lots of work to try to get the word out there.
Well, but let's go back to your previous question. I interrupted you again. I'm sorry. with the finger example. So how would you use it? The thing is, most people tend to want to make the investment because they got a problem they're dealing with. And most of the people that I work with, and I do consultations, so anybody wants advice, specific advice on their particular situation clinically, then I will do a half-hour consultation and then provide you advice on the best system for you to have for your problem. Constellation of problems. Rarely is it one problem, mostly it's multiple problems. So whole body systems are the best because again, you're doing prevention. The biggest investment is the control unit. And then you have a whole body system and you can have a smaller applicator that you can apply to different parts of the body separately.
Okay.
Right. So you make it an adequate investment, and then it's a question of how much do you use it. And there's no good answer for that. So I tell people I can't look into your body. I don't see what's going on at the cellular level. So if you have what is it, 100 trillion cells in our bodies, and every cell has about 2,000 biochemical processes per second.
Yep.
How much should you be using it?
Right. Exactly. Well, what do you find in your years of experience is effective? I mean, 15 minutes lying on it?
No, there are companies that are selling magnetic therapy devices that are 1 Gauss or less. So anybody buying a PEMF system, if they don't know what the intensity of that system is, don't buy it.
Right. Okay.
If they're not willing to tell you what it is,
then don't buy it. For sure. Yeah.
Right. So always ask, what's the intensity? Because the intensity drives the inflammation. We talked about the adenosine receptor. You need 15 Gauss optimally.
Right.
So if the system is delivering only 1 Gauss, it doesn't matter how much you use it.
Right.
It's not going to do much. It'll still help. It'll still make you feel good because it's stimulating the acupuncture points.
Right.
But they're not going to do much healing. Time is a problem for everybody.
Yep.
So I think the more motivated you are, the more time you have available, the more time you're going to need. So typically the answer is, particularly for pain, if you're doing a treatment and the pain comes back an hour later, then retreat. Now, maybe it is only 5 minutes or 10 minutes, but if you did an hour treatment and the pain took 8 hours to come back, that's probably better than a 15-minute treatment. So physiologically, people are going to have to find out for themselves how much time they really need to get the best results.
Okay.
My usual recommendation, even for health maintenance, is a half an hour twice a day.
Oh, okay. So that's a substantial time commitment. I guess you can be doing other things, meditating, whatever, at the same time.
You can meditate. It's hard. You can read. You can, with a smaller applicator to your back, you can still work at a computer.
Whatever, right?
Yeah. But this is the point. People have to decide for themselves what benefit they want.
Right.
Considering the investment they made. If you can get by with 8 or 10 minutes a day, fine. What happens is at the beginning, you're going to need more time to get everything tuned up. Get everything that you can optimized. And after that, all you need to do is to maintain. And if you back off your maintenance level, if you back off your therapeutic level back to maintenance and the problems reoccur, then you haven't done the job yet. You haven't healed the tissue.
Okay.
So everybody's going to find out for themselves how much time they're really going to need.
And I'm sure our listeners and viewers can go to your website to get more details, but just generally, what are we talking about for a good system cost-wise? What would be the range?
So because intensity matters.
Yep.
So some of the 1 Gauss magnetic systems that people are being sold in multi-level marketing programs are about $5,000 to $6,000.
Oh, wow. Okay.
Okay. That FlexPulse device I mentioned to you is about $1,000.
Okay.
Right. So but that's a small local device. If you want a whole body device, it's going to give you enough intensity to help you deepen your body with healing purposes. You're going to be talking minimum 3,500 Gauss, probably closer to 6,000.
Okay. And it's a pad you lie on, basically?
Yes, it's a pad you lie on. It's got a small control unit, and then you plug an applicator in and you lay on the pad, and then you can unplug that applicator, put in a small one to treat a spinal stenosis or whatever else. Okay. That's what you should expect to invest. Again, it's an investment.
Sure.
Right? And it's going to be an investment in time as well. But when we are dealing with anti-aging, what do we need to do to
we want to live longer, healthier. Yeah, absolutely. So, I mean, that's not a crazy investment. You're sort of along the lines of other types of things, some of the portable sauna type things, or the infrared devices, et cetera.
Portable HBOT,
Yeah.
or
Mhm.
or portable light systems. You can spend $3,000, $4,000 for those.
Sure.
And the problem with those is they don't penetrate the body deep enough.
So you feel like if you're doing the PEMF, you don't necessarily need to do the light therapy for photobiomodulation?
They combine, but everybody's got issues with resources and time, right? So I found over 30 years of doing this and having used all these other devices as well, magnetic field therapy is the best value because it reaches complete through the body.
Okay.
Right? Most of these other systems are limited. They're very shallow.
Yeah, they don't go that deep, right? Exactly. If you're looking for skin, fine, but otherwise, yeah.
For the brain, for example, you're going to need 4,000 Gauss to go from one side of the head to the other.
4,000. Wow.
Gauss, yes.
And
Deliver your 15 Gauss optimal intensity.
right. Oh, I see. Oh, okay. So, and there are devices that do that.
Yes, the devices, the ones that I mentioned, especially at the $6,000 level, are going to do that.
Great. A little thing just popped into my head before we end, and you tell me whether this is silly or not, but with all the advent of the electric cars, is there going to be an issue with PEMF, people driving around in these electrical vehicles? Not PEMF, EMF.
Yeah, so EMF. I'm going back to the original discussion about EMFs in general. Even radial tires produce an EMF.
Oh, right, because they have some metal in them and they're spinning.
Yeah, well, they're bending and
Right.
constantly changing. Now, people have done measurements on the EMFs produced by a battery in an electric vehicle, and it's very small.
Okay.
Now, if you're going to be driving long distances, if you're doing a commuter and you drive 15 minutes or half an hour, it's not a big enough dose. Because the dose is relatively small and the time exposure is relatively small, your body, like any stimulus, like a cloud passing over the light, or the light shining through a cloud system, that minimal exposure is going to stimulate the body to some extent, but the body will recover. So, for example, a healthy person, when we did brachial artery studies on with magnetic fields and looked at the vasodilatation of a brachial artery, and again, healthy people, and what they found is that the artery dilated probably within a microsecond. Dilated just as rapidly after the stimulus ended. It vasoconstricted for about the same amount of time and then went back to normal.
Oh, okay.
So all of these EMF exposures create a physiologic response. And then the body has to be able to recalibrate itself. So that's homeostasis, right? So it's going to change. The body says, I don't want to be dilated. I want to be this. So it compensates, cybernetically compensates, goes back in the opposite direction, and then goes back into neutrality. So all of these kinds of exposures, if they're not very high and over extended periods of time, most of the time physiologic responses are going to rebalance. Cell phone use, Wi-Fi use, cell phone towers, all of that are small doses for most of us most of the time over a long period of time.
Right.
And when you're doing magnetic field therapy with good magnetic fields, you're constantly rebalancing the body. You're stimulating the body's natural systems to rebalance themselves. So you can, to a great extent, undo the physiologic imbalances you're getting with the wrong kinds of magnetic fields.
So it sounds like something we all should be doing on a regular basis to rebalance that.
Absolutely, because again, there's no minute in a day that you don't have to rebalance.
Yeah, for sure. Well, this has been a really fascinating and comprehensive discussion. Opened my eyes up about the role of PEMF in health and disease. I really appreciate you taking the time to talk to us. Dr. Pawluk, it's drpawluk.com, drpawluk.com, which is, I looked at the website, fantastic information on there, and you can get consultations as well. If you want to put your book up, the latest book up for them to take a quick look at as well, so readers can get that.
And we're going to provide your listeners in your show notes. We give you some links. One is to get the list of 80 conditions that we have in the Supercharge Your Health book.
Great.
And number two, if you want to order the book, you can order it through that link that we'll provide you. And number three, if you want a consultation, So if you're not serious about your health, I'm not going to spend half an hour trying to convince somebody to buy a $5 system.
Right.
Right.
Sure.
You have to make an investment in your health. So if you really want to make an investment in your health, then we'll provide you a free consultation.
Great. Fantastic. Well, I may well be giving you a call.
Well, you don't have to. If you want to call, we'll have a call offline.
All right. Very good. Thank you again. Pleasure speaking with you.
Likewise. Thank you. All the best at your summit.
Thank you. Before I bring up our next speaker, please make sure to share this information with your colleagues by sending them the link to watch. Welcome to another episode of the Telomere Summit of the Dr. Summits. I'm your host, Dr. Joseph Raphael. I am very happy to be here today with Dr. Christopher Shade, who is going to be talking about his work over the past few decades in detoxification, mercury systems, and then he'll get into his longevity wheel and its effect in detoxification, and we'll talk a little bit about telomere biology as well. Welcome, Dr. Shade.
Thank you. Pleasure to be here.
So, Dr. Christopher Shade is the founder and CEO of Quicksilver Scientific, continues to be a driving force of development and innovation. Dr. Shade's vast depth and breadth of knowledge, passion for healing, and intuitive understanding of chemistry and biology are reflected in Quicksilver Scientific's well-designed detoxification protocols, unique supplement delivery systems, and patented mercury speciation test. Dr. Shade is a recognized expert on mercury and liposomal delivery systems. He has lectured and trained doctors in the US and internationally on the subject of mercury, heavy metals, and the human detoxification system. Dr. Shade's current focus is on the development of cutting-edge lipid-based delivery systems for nutraceuticals such as liposomes and microemulsion systems to address the growing need of high-quality, affordable detoxification solutions. Well, let me just start, as I do with many of my guests, because I'm sure you have a fascinating journey to get to where you got in this field. What started you out in this? What kind of training did you do? And tell us a little bit about your journey toward to where you are now.
Yeah, it's kind of a funny and long journey. I'd always had a lot of interest in health. It really began in college. I didn't think about it a whole lot. And then I had this sort of break from, what I was studying, which was environmental science. And, I realized they were just sort of helping the polluters. And I took this kind of radical break and I went into organic farming. And I joked that I went out of business as an organic farmer the year that Whole Foods came around. So I was a little bit early. And then I studied some organic farming research. I worked for Rodale Institute, and they had this motto: healthy soil, healthy plants, healthy people, or healthy soil, healthy food, healthy people. And, I was looking at it that way. And then I went to graduate school and I got a PhD, and I was looking at what's called biogeochemistry, and it was mercury biogeochemistry. So elements cycling through the environment, how they transform. And a lot of these are toxic elements, and here we're looking at mercury becoming a toxin, building up in the food chain and thus, and got my PhD, and I finished some testing for different forms of mercury where we could separate the mercury from dental amalgams from the mercury from fish and look how it's compartmentalizing in the body and how it's starting to come out. And I brought it over into integrative and functional medicine, and I realized they really had no idea what they were talking about. I mean, people were doing detox and they were using chelators, but their understanding was very unsophisticated compared to what it was in environmental science. Now, I think this is because no money was put into clinical mercury science, really because of all the controversy around thimerosal and vaccines. And so the government shuttled all this money into environmental science. And we knew everything about mercury going through phytoplankton to zooplankton to fish, where it compartmentalized, where it goes in the cell. And so I wanted to apply some of that and get us away from this sort of heavy-handed approach to mercury in, chelator, mercury out, to, how does the cell supposed to deal with this? And so I focused a lot on the glutathione system and how that gets turned up, the transporters, the transferases that go with it, things that link toxins like mercury onto glutathione, how they get shuttled out of the body. And that led me to these big gene switches, NRF2 and AMPK, that work with that. And, frankly, in the beginning, I started coming to A4M and doing anti-aging shows back in like 2010. And, I was a little jealous, they're doing like stem cells and, telomerase activation. And all I'm doing is taking out the trash. But the farther I got into it, the farther I looked into it, the more you see that, it's a very deep biochemical picture when you're cleaning things up. It's not just cleaning up environmental trash, but a lot of this is metabolic trash. We're making proteins all the time. We're misfolding them. They're building up. They're becoming inflammagens. They're crowding the cell. And we see there's this rhythm of anabolism and catabolism, and we need to sort of link those two together so that during catabolism that's activating AMPK, and NRF2 wants to go with it, and we clear all that stuff out of there, and we tune up our antioxidant system at the same time. And guess what? Things like telomerase are activated when glutathione's high and it's highly reduced, when all these inflammagens are getting out of the cell. So over time, I sort of built up this idea of the longevity wheel. What are the things that we have to hit? There's NRF2, AMPK, NAD, sirtuins, telomere attrition and telomerase activation, senescence, dealing with that, and the neuroendocrine system. And we see they all go together, and sort of all those paths lead back into this cellular cleanup and this maintenance of a clean redox poise in the cell. And, it really goes back to these old, 100, 200-year-old ideas of a clean terrain, both outside the cell and inside the cell. And so as we go into talking about the Telomere Summit, it's natural for me to talk about detox because, over time I built up my whole company around developing these nanoparticle deliveries to support that, but then also support these higher order metabolic things like the sirtuins and the NAD level. And so that's where Quicksilver came to be back, around 2009, 10, and we built up now over the years to, making our lifestyle on this.
Really interesting. So let me just step back a little bit and talk about specifically mercury and how it was interesting to hear, because I know if you talk to the toxicologists at the state laboratories, they're like, well, we don't care if the mercury levels, as long as it's under 50, we're good. Then you get the call. But then you talk about the integrative physicians who are using chelation stimulation to see whether or not you have a mercury overload. But what you're saying is you have a way of looking at it without having to do stimulation and then separating out methyl mercury, which is organic mercury that you measure in the blood, from inorganic. Sometimes people get that confused where, let's just get rid of mercury. Well, maybe you have a lot of one kind, but not a lot of the other. Tell me about what your, how you separate those out, and then we can get into how that starts to affect the body.
Yeah, so we use something called mercury speciation testing. This is what I did my PhD on, was to have one method where all the mercury forms go in, but then we chromatographically separate them out and measure them individually. We do this in blood, hair, and urine. So toxicology would only measure total mercury. And if you're exposed to inorganic mercury, they'd look to the urine. And if you're exposed to organic mercury, they'd look to the blood. But what we found, once you separate these two, urinary inorganic mercury should be proportionate to blood inorganic mercury. And when it's not, that shows that there's some damage to the transport mechanisms that are in the proximal tubules of the kidneys. So you've got, some glomerular damage in there. And that's one of the easiest places to scar up, especially when you have metals and endotoxin together. So you got a bunch of amalgams and you got leaky gut, maybe periodontal disease. You can really knock out the kidneys, and you can build up a large amount in the blood, but a low amount in the urine. And in fact, when people do the chelation test, if you have this kind of transport damage, you'll take the chelator and you won't get much out in the urine. You'll get a false negative. So that was a really nice thing to have those two independent. And then the organic or methyl mercury in the blood is related to what comes out in the hair, and they should be linearly related. When they're not, this one's not as direct, but it reflects more of a disorder in cycling related to glutathione and liver processes. So we've got a load of what's in the blood, a load of what's in the body and the blood separated into the two forms, the organic from fish, the inorganic from dental amalgam, and the excretion paths in the hair and the urine. Now, this story had always built up that the chelator was going to show you your body burden, which you couldn't get any other way. Like the chelator would go into every cell and take a representative amount and dump it all out so you knew what was going on. Well, that was a story that was built up because they don't go even into the cell. They don't across the blood-brain barrier. They're taking proteins that are in the blood and lymph, solubilizing the mercury that's protein bound into a water soluble kidney filterable form. So they're taking what was already there, but making it a lot easier to see. And so 30 years ago, when they started doing this, you just couldn't measure very high in urine and blood. It was just like not sure what was, it didn't relate to amalgams. So they kind of made up these stories around there. So once we had really good analytical technology that could measure down into the parts per trillion level and separate the different forms, the whole picture's right there for you. And so that was the move forward that we took in the analytical. And then in the treatment, it was, all right, how do we get it out? Well, it's up in the tissues, it's in the cell. How do you get it out naturally? You link it to glutathione. So we got to get glutathione in there. We need to have the transferase that pulls the mercury off the cellular proteins and links it onto the glutathione. And then phase 3. So people talk about detox in phases. There's phase 1, phase 2, phase 3. Phase 1 are more oxidative reactions to take non-reactive things like flame retardants and some of the pesticides and herbicides, make them a bit of a radical, and then link them in phase 2. All right. And that's, when Jeff Bland talked, it was phase 1 and phase 2 hepatic detoxification. But then phase 3 is like a relay race out. And so they're transmembrane transporters that actively move this toxin conjugate out of the cell into the blood. From the blood, pull it into the liver. From there, dump it into the bile flow. So all of a sudden, it's like, oh, wait, we got to do all this stuff in this cell. We got to dump it into the blood. Then we need the liver to harvest it and coordinate it with bile flow, because actually the toxin transporters out of the liver are also bile transporters.
Right.
So anything that blocks bile flow blocks toxin flow. So that was our movement forward, to get all that harmonized and then make sure you have the proper binders in the GI, specific ones for metals or mold toxins, to catch all that and get it all out to fecal excretion.
What are those binders?
So for metals, we use something called IMD. That's a proprietary thiol functionalized silica. So the chelators, the bloodborne chelators, DMSA, DMPS, those will have dithiols onto a central carbon molecule that's negatively charged, and they'll make these complexes you pee them out. So we take these very high surface area silica particles and covalently bind on carbon chains that terminate in thiol groups. So you've got just millions of thiols coming off every little particle, and those go through the GI, and as those metals come out with glutathione or metallothionein into the bile, they then grab them and hold on to them so that you don't reabsorb them. And the most famous of the reabsorbing metals is methyl mercury. It's got about 95% reuptake. Cadmium's got a lot of reuptake, a little bit for arsenic. So that just ensures that you take everything all the way out.
So
then when you're talking about mold toxins and more environmental toxins, using a combination of charcoals and zeolites and chitosan. Chitosan is analergic but shellfish derived. That's repeating amine groups. It's analogous to Welchol or cholestyramine.
Oh,
a
okay.
little bit weaker than cholestyramine, but similar to Welchol.
So you don't use any of these actual EDTA type or DMSA chelators?
The one that we use is EDTA. We do a liposomal EDTA. Mercury, you got 4 main metals. You got mercury, cadmium, arsenic, and lead.
Right.
Mercury, cadmium, and arsenic are very thiol reactive. They get linked onto glutathione and get out of the body. You can take them all out with a glutathione system upregulation. Glutathione system upregulation will block the toxic effects of lead, but it won't export it. And so there, some EDTA is very effective. DMSA is effective there too. And a lot of people, like Mark Hyman's group, they'll use our system as a base, and then they'll top over top of it, a little DMPS or a little DMSA or EDTA in various forms. But within our system, EDTA and a liposome is the only one of the chelators that are part of the system.
And how do you follow the effectiveness of it? Laboratory testing?
Yeah, we're
The same
doing
thing. You just
the
follow
blood
your test
testing.
and see it come down?
Yes, the speciation testing before and after, and making sure everybody's come down. We had a. Do you know Neil Nathan and Eric? I forgot the guy who worked with them now. I forget his last name. They ran a big clinic out in Santa Rosa. They've been doing DMPS chelation for 20, 30 years. And they did a whole thing comparing ours to what they're used to seeing. And they said it's as good or better. And the beauty of it is you're getting the body to do what it's supposed to do. And so turning that up through this trigger, NRF2, is turning up the intracellular formation of the antioxidants and all these molecules. And in fact, if you're doing chelation with DMSA or DMPS, you're not getting into the intracellular stores. The only way they can get out of the cells is through NRF2 upregulation, because with NRF2 comes synthesis of glutathione. We bring in liposomal glutathione to just help that, but NRF2s bring in synthesis of glutathione and activation of all these enzymes to dump things into the blood. In fact, if you really strongly upregulate NRF2, you'll see the blood levels come up for a little bit as the transport mechanisms clearing the blood are trying to keep up with it. And then when you stop, it'll go down below where it was before. So at some point you need, to turn up, squeeze the sponge of the cells, get them into the blood.
Just for our listeners, talk a little bit about the NRF2 system and kinds of things you use, I guess, sulforaphane and other things like that for upregulating it and how important it is.
Yeah, so NRF2 is a stress response trigger in the cell, and it's located in the cytoplasm. And there's a pair of proteins, NRF2, and it's held in place called something called KEAP1. KEAP1 has these. KEAP1, KEAP1,
okay.
Like it keeps it in place. And so it's got these sensitive dithiols on it. Dithiols are very sensitive to oxidative damage or electrophilic damage. Electrophiles are like reactive oxygen species in that they pull away electrons, electrophiles. But that would be any of the metals, various environmental toxins, mold toxins. And so if you have a buildup of chemical or oxidative stress in the cell, you oxidize these dithiols and it releases NRF2 and it goes into the nucleus and it activates transcription of a family of genes that has a promoter region called the antioxidant response element. And this family of genes includes intracellular antioxidants like glutathione, like superoxide dismutase, thyroidoxin, and all of the things that go with them, like the transferases, the transporters, different things for cleaning up old proteins, molecular chaperones, and all these clean up the cell and lead to cell survival under these stress conditions. Now, over age, just like telomeres go down, the activity of NRF2 goes down. So NRF2 is inducible, meaning under stress it induces, but it also has a rhythm with which it naturally goes in and cleans up the cell. In fact, all these proteins, they used to be called house cleaning proteins before they subsumed them all within this whole chemo protection system. And so over time, this rhythm with which it gets induced goes down. But we can bring things into the system which induce it to turn up transiently. And these are things like our lipoic acid, sulforaphane, in fact, quercetin, and we'll hit on quercetin a bunch of times in this talk because it does so many different things for us. So the general classes, you've got the sulfur molecules like the sulforaphane, isothiocyanates like you get from wasabi, and then lipoic acid's the one I use the most, and then the polyphenols. So the polyphenols would be like EGCG, haritaki extract, quercetin, resveratrol, and curcumin to a lesser extent. All of these have this effect And interestingly, we think of these as antioxidants, like, oh, lipoic acid, antioxidant, green tea extract, antioxidant. They're actually little free radical generators, but they don't have collateral damage. And they're light but effective at changing that conformation of KEAP1 so that NRF2 translocates and upregulates all these good genes.
Right. Well, that's a great explanation. And so you try to upregulate these systems. Maybe we should talk a little bit about your longevity wheel sort of protocol.
Yeah. So, it kind of looks like a Star of David. There's these 6 points around it. And at the top is NRF2 AMPK. And we're going to start there for even a couple of months. And like we have a program now, we call the BioAge Reversal Program. And we took 44 people through this program, and we did true age diagnostics, epigenetic age clocks on them before and after. And we've done some serum markers as well. And in the first month, we're focusing on NRF2 with glutathione, and we use a little cat's claw to try to address some of the latent viral activity and trying to turn up the cleaning up of the system. And we do it in this, we do the NRF2 upregulators with the bioflow modulators as a dose, and it's something called liver sauce. And so you've got traditional bitter compounds there for, yeah, I know, great name. It's like A1 sauce for your liver. And so it's got traditional bitter compounds like gentian and myrrh and dandelion root to turn up bioflow, along with these polyphenols and sulfur-based upregulators, NRF2 and AMPK. And in the nanoparticle format, that gets in and gets absorbed and peaks in the blood between 20 and 30 minutes. So it activates all this flow, couples it to bile, and then a half hour later, we come in with a binder mix, the thiosilica, the charcoal, the clay, the chitosan, and carry that all away. So we do that for a month. Then we switch over to doing the same, what we call push catch, like activate and then bind. But now it's a little bit more AMPK focused, and we'll do that for another month and add in NAD precursors. We use nicotinamide mononucleotide and a liposome and some membrane builders. So in this longevity wheel, the mitochondria is sort of the center of the focus, and the mediators of everything are the membranes on the cell membrane, the mitochondrial membranes, the endoplasmic reticulum, the Golgi. They're the communicators that talk about everything, and they get all damaged by the reactive oxygen species, the toxins. And so we're moving out toxins, and then we're trying to activate AMPK. And this is a big one these days. I talked about it at A4M, and David Perlmutter followed up right behind me talking about it as well. And AMPK is pushing us into lean metabolism. It's what happens when we water fast, when we carb restrict, when we do keto diets, when we exercise. It's saying, oh, we're using up the available energy. Let's mobilize fats and make ketones out of them. And thus we're going to mobilize fat-soluble toxins. Let's mobilize glycogen, glucose. Let's turn up glucose transporters and go into a lean metabolism. And so we do that for another month, and then focus another month on membranes and NAD. And so this is to get the poise of the system back really tight. And it's so effective when you do these things. In fact, just that first month protocol, we had a guy named Cheng Wan use this system for looking at fatty liver, and he got 82% resolution of fatty liver in 1 to 2 months just doing this kind of a system. And
I interviewed Dr. Wan. Dr. Wan.
At the time he did 100 patients, and he's got 800 that he's ran through this since then. And so we're going to get that together and publish it. So you see, this ability to move into clean metabolism, and one of the things, when the toxins go in, they'll hit the telomeres, and there's different toxins that do that, the most, and probably cadmium's the worst. Arsenic and lead right behind that. Tobacco is really renowned for that. I think as we look more and more of these, even if we look at like mold toxins, we'll find there's a lot of telomere attrition from that. leading to mitochondrial dysfunction, leading to cellular senescence, leading to the propagation of those spreading fields of inflammation that are recruiting everything else into that. So we want to be able to, when we're choosing these compounds, be able to address that already accumulated damage and, that load of senescent cells that we have. And in our choice of the compounds, we use a lot of quercetin because it's a good NRF2 upregulator, great AMPK activator, stimulates mitochondrial biogenesis, and it's a senolytic as well. And so we did find at the end of this 3-month period that the beta-galactosidase, which is it's a marker of cells that are releasing it, meaning you're breaking down senescent cells, that the beta-galactosidase had gone quite a bit up. And then we want to follow up after we stop it a couple of months and see it come back down.
Did you measure that with
really
Dr.
exciting.
Did you measure that
with Dr.
Yeah.
Yeah, with Ginfinity, and a lot of these things, we're still working out what's ready for prime time and what's not,
Right.
NAD, where are we going to measure it? What does it mean to measure it in plasma? Because it doesn't mean the cells are high or low, but it means
Right.
something. What does it mean to measure it in red blood cells? And do we have that assay wound up? Where should we measure AMPK? Certainly not in plasma, but maybe in the PBMCs.
We're in the early days for sure. Yeah, we're definitely in the early days on that. You mentioned TrueAge.
Yeah.
Did you get pre and post?
Yeah, we did. We got pre and post. We got a nice, very highly significant reversal on the new Dunedin-Poam algorithm, the original Horvath algorithm. And they've got another one, I think, out of Yale they're going to run on that now. And that's in the early days. Okay, we moved that back. What are the genes affected? Well, I don't really understand them, and so digging into these and which ones really mean something. But one of the really nice things, we're in an age of, immune challenge. And so we saw a nice reversal on natural killer cells, T cells, and beta cells. And at least in their epigenetic profiles. And
so you did the
we don't realize
extrinsic aging as well with him. You did that. You did the intrinsic, which is the Horvath TrueAge, and then you also did the extrinsic one, and you saw some changes in the T cell subsets based on their DNA methylation pattern.
Yeah. Those are really nice. It was B's, T's, and natural killer cells, and so with that, AMPK, one of the things that, NRF2 and AMPK both relate heavily to immune function. Glutathione levels, and specifically the amount of reduced versus oxidized, are very strong modulators of immune response. When glutathione goes down, you shift into like Th2 and Th17 type of inflammatory activity and away from your Th1 activity. In fact, interferon goes way down, and then you bring the glutathione back up and the interferon comes back. And then with AMPK activation, you have activation of STING genes. Those are interferon genes. And the process of autophagy comes with AMPK activation. We know that more for mitophagy and endoreticulopathy, taking old mitochondria, breaking them down so that you can make new ones. But that autophagosome that's produced in autophagy, and then it's linked together with a lysosome to break it down. When you do that with microbes, virus, bacteria, parasites, that's called xenophagy. And it's allowing you break it down into subsets so that you can form the right antibodies to it.
Oh, okay.
And so, so these are huge elements of our immune response. And then like glutathione, its relationship to telomeres. The higher the glutathione, the higher the telomerase activity. But it's not just total glutathione. You've got to be shifted. The more reduced versus oxidized, the more telomerase activity. So that all comes from glutathione synthase and glutathione reductase, which are subsets of what's upregulated during NRF2 upregulation. So we see a lot of these things tying back together. And yes, we got significant changes there. And the phenotypic changes are amazing. I mean, looking at changes in fatty liver composition, looking at changes in energy, brain clarity that come with these things, and as you said, with the Genfinity analyses, we're just getting into what should we really be looking at with all these things and what compartment should we be looking at them.
Yeah, so the couple of studies, the TRIM trial and Kara Fitzgerald's study recently published results on epigenetic age reversal. Was your change in, do you have a number? I'm just curious about.
Yeah, no, I just got these results back from them from this 3-month trial, and so I'm going to meet with Ryan next week, and we're going to go deeper into them, and and then I got to break open all 44 people and say, oh, this guy went 3 years back, and this guy went 3 months back.
Right.
That guy
so
went
it's
forward.
Still early. I didn't know how long ago you'd done it.
No, we just got this stuff back, and and so we have to translate these into amounts of time. And then we want to go through the second part, because the first part, you're kind of stirring up the pot too. I'm going to move all these fat soluble toxins out. I'm going to try to get them out before they do any damage, but you're going to have a little bit of back and forth going on there. And then you get into the second level of this, and there you're just feeding NAD, being, light activate, feeding NAD, feeding sirtuins, feeding neuroendocrine, bringing in pure astragalosides, different high-end adaptogen ginsenosides, and looking to drive the whole thing forward. So, give them another 6 months on the nurturing side and look at them again now that we have that baseline data. Their 3-month intervention to clean up and restore the terrain, and then 6 months to build it back up and see where we go from there.
So, and just in your overall protocols, what types of doses are we talking about for alpha lipoic acid and for NMN and those sorts of things? I mean, I guess since they're in liposomal systems, it may be that they're lower than you might expect.
Yeah, sometimes you look at, like, when we're doing quercetin in the AMPK activator.
Quercetin, sure.
40 milligrams of, 40
That's
milligrams of that.
Yeah.
Yeah, and then you do the pharmacokinetic study, and it's a 25-fold increase in bioavailability.
Right.
So, now your 40 is a gram, and it's happening twice a day. And the other thing is how much you focus it into a window. You're going up and you're peaking between 20, there's another peak at 40 minutes, and in 90 minutes, you're out of the system. And it's these very high levels that are really inducing, these are all inducible things, NRF2, NPK, sirtuins. And so having everything focused in at once and having a bunch of overlapping ones. So in the AMPK product, you're bringing in quercetin, resveratrol, berberine, silymarin, and DIM, all hitting same targets and different targets. And so that's where you're really able to generate a lot of work. In fact, a lot of people, you see them go from subnutritional ketosis into ketosis, a nutritional ketosis in 30 to 60 minutes, because you're able to hit AMPK from so many levels. So you do that twice a day with the binder systems. When we're bringing in NMN, we're bringing in 100 milligrams twice a day. We're looking at we had to do CACO2 studies on that because they're harder to get a good blood measurement off of. And that looked like about a 4-fold increase in bioavailability. So, the insolubles tend to get larger increases versus the water solubles.
Sure.
You can also get more into the particles. So, as we go, we're refining, we've got ICP-MS, LCMS triple quad in here. So we're trying to get all the bioavailability equivalencies. And one of the interesting things, when you get them in nanoparticles, you don't have them metabolized. So like curcumin, unmetabolized, non-glucuronidated curcumin in large amounts, and then you see the glucuronidation come up. And a lot of these are much more biologically active before they go through that phase 2 metabolism.
So this is a proprietary liposomal sort of formulation?
Yeah. Yeah, we're making either liposomes or lipid nanopart or nanoemulsions and sometimes microemulsions. Everything's sub 100 nanometers. We make everything in house. We've got 1 patent issued, 9 patents pending on different forms of this. We do even things like making emulsions for Molson Coors' cannabis systems, cannabis beverages in Canada. So we're putting this technology into a number of things. Always trying to keep particle sizes a certain size. We have a lot of controls on that system, and just trying to advance bioavailability so that we can get these like really powerful results. We know what the what the potential of these small molecules is. Everybody in the research world knows that natural small molecules have great potential, but bioavailability is what holds them back. And so we get rapid and high blood levels in, and then we get the changes from those. That's how it's able to, move things like adiposity in the liver in, 1 to 2 months, just through getting so much in right away.
At the same time, you're measuring such sort of more conventional things like hemoglobin A1C. You see kind of major changes with that?
Ironically, we hadn't been tracking that all that much, and we just started doing oral glucose tolerance tests, and we see people coming from peaking up around 180 to staying on 135, 140.
Nice.
With those AMPK activators. So that was really nice to see. And, as we move forward in the beginning, you don't have a lot of money, to do these larger trials. And now we're trying to bring in, other groups and get larger trials going and, just fill in all this data sets. Originally our strategy was everybody knows what the molecule does. We're just going to go in and show we get more in. And then as we grew the company, have more money to throw at it, now we're doing these outcome tests.
Well, that's important, and I think the supplement industry in general is starting to realize that, because there's starting to be overlap between pharma and the supplement industry, because natural molecules are sort of where it's at, and if you don't show that same, not just pharmacokinetic data, but efficacy data, you're not
pharmacodynamics and the endpoints on the studies, that we're getting there.
I'll be very excited to hear about what the epigenetic age reversal was. Do you do any telomere testing?
We haven't yet. We do want to start doing that, and look at blendings of different things, adaptogens and the astragalus products at the same time, along with all these. And so there's various intellectual property around that we got to be careful of.
Oh, sure, yeah.
So get TA to let us do it.
Well, yeah. I mean, you could. I guess you could put the molecule in one of your overall systems.
Yeah,
that would be interesting.
Yeah,
or giving.
Yeah, I think that would be a great way to go with it. And I'd love to team up with them and get some of the data around that.
Yeah, because it might well be synergistic, and you'd see. I'm sure it would be synergistic, and that would be helpful. Well, I mean, it sounds like you're on a real pathway towards a very comprehensive approach to age reversal. Let me ask you, are there any other things that you want to talk about, tell us about that you're involved in before we wrap it up?
No, I mean that about hits it. Except we just got into hormones, and that's been a lot of fun. We did a female system over the counter with nano DHEA and pregnenolone and some modulators like chrysanidin and some adaptogens. And we found, in the nanoparticle, because it comes in unsulfated, it can go into testosterone like that. And so it's been really great watching even middle-aged, pre-menopausal women and then menopausal women, refilling their baskets with that. And for men, we did a nanoparticle testosterone that's going out through college pharmacy, and we're really interested in
as a transdermal.
No, as a sublingual. We did transdermal progesterone, which works great, but the sublingual will take you, say you're running like 300-400, take 12 to 15 milligrams, you'll go up through 2,000 to 2,500, and then come back down to baseline over about 3-4 hours. And the idea is to go up activate the androgen receptors and get down so that you stop suppressing LH and FSH so that your testicles still make the natural amount they're going to and do all the things that they're going to do, not have the testicular atrophy.
It's really
And
quite,
this is incredibly.
yeah,
Go
that's
ahead.
really quite similar to Natesto, I guess, essentially the nasal delivery system that pharma has.
Oh, they have a nasal. Yeah, exactly. So it'll come in and then go away.
Two,
And
three
so
times a
especially
day.
in the yeah, and especially in the younger men who've
Right.
had maybe TBIs that are lowering their testosterone, to give them the androgen activity without having the contraceptive effect of doing like constant cypionate injections.
I'd be interested to see if because I mean, they have micronized progesterone and you've used a transdermal form. I'd be interested to see if,
Yeah.
I don't know, you would be able to speak to this more than I would. You're talking about nanoparticles. That's, I presume, smaller than a micronized progesterone. Could you
Yeah.
potentially get a more effective progesterone at a lower dose?
Yeah, we're using 8 milligrams right now, and it seems to be enough. And when
Really?
you look at the peak, yeah, you
Wait
look
a
at the
second, 8
PK,
milligrams
like
of progesterone
yeah,
in a
and,
transdermal?
yeah, in a transdermal, some people will double that and stuff, but it like really chills you out and makes you sleep. And when you watch there's a peak in about 100 minutes that goes up to about this in that PK we did 20 milligrams and it went up to about 20. I forget, is it nanograms per deciliter?
Nanograms per
And,
ml. Uh-huh.
yeah, it came down and then went back up and came down and then went back up. And then after about 12, 18 hours, it's back to running, about 1. But it goes transiently. So we have people put it on at night. It goes through some really high peaks and really works well. It's truly dissolved and it's in these nanoparticles that are about 30, 40 nanometers. You do that topically and they really run in. So we'll do that with tests too. But we did the topical because we're a supplement company and we can't do a sublingual. We have a sublingual, and there's people who have already been using that for changing endometrial. There's a guy treating endometrial cancer with it, and the sublingual has to be gone through pharmacy. But the topical really blew our minds.
So, I know the other reason you're using that such a low dose is because you can use up to 20 milligrams topically, right? I think that's the magic number.
You can use up to 2%, and
Right, 20
that
milligrams,
tends to be up to about 20 milligrams. But 20 milligrams, so people who are doing 20 milligrams every day, we're too tired.
So when you're measuring those peak levels, is that you're using LC tandem mass spec to measure those? Is it actual?
Yeah, we just send
or?
them into Access Labs and let them
okay.
run.
You send them into Access Labs.
And we couldn't even do saliva because it blew out the ranges.
Saliva, that's a whole nother discussion. But
in
terms of blood, I'm just curious because I do a lot of hormone optimization, of course, whether or not you can get actual progesterone levels that high, or what your metabolite profile is, because the progesterone is metabolized into many things, and a lot of them are active, like 17-hydroxyprogesterone, and then the sleepy component is the allopregnanolone that you get. So if you can do it with such a small amount of progesterone, that's really interesting. And usually you don't get the sleepy effect when you use it topically. You more often get it when you use it orally. So I'm curious about that as well. We should talk a little bit more about that.
It's stony and sleepy, and there is just no doubt. And everybody's like, man, that stuff's great. So we're going to do metabolites with Frank Nort from Rhine.
Okay.
He does 24-hour urine collection and does all the metabolites. He doesn't dry it down. So we wanted to find out more what was going on. But it was really relevant blood levels, for a couple hours after the dose. So, we had stopped at 6 and it was up at 20. And, between there and, 12, 18 hours, it comes back down. So you could do more than once a day if you want to keep it up there. But, it really does the trick.
Well, very, very interesting. Well, I'm glad we talked about that. It sounds like you're doing a lot of really exciting work, advancing the field, and it's been a real pleasure talking to you about all this stuff and educating our listeners about it. Is any stuff you want to tell our listeners about social media or your website or anything?
Yeah, quicksilverscientific.com. Go there. Doctors get a doctor account. You'll have access to all of our educational resources. I've done so many webinars. I do about 1.5 hour webinars. I think we have 40 or 50 online for the doctors to access. The consumers can get accounts and get access to some of our educational material. We do some direct to consumer on social media. There's Dr. Christopher Shade on Instagram. You can tell I'm not a social media guy. There's Dr. Chris Shade. I do a podcast series, and that's a website, and then Quicksilver Scientific on Facebook and Instagram to follow what we're up to.
Well, great. Well, thank you very much for taking the time to speak to us, and look forward to seeing you at one of the conferences soon.
Absolutely. Thank you.
Are you the type of person who likes to learn at their own pace? Plus, you're bound to miss something with so much wisdom being shared over 7 days. When you upgrade to VIP All Access, you won't miss a single second of this incredible game-changing summit. You'll have lifetime access to all of these expert interviews and all the bonuses to watch, listen, learn from, and even read the transcripts whenever and wherever you choose. And don't forget, upgrading to VIP on demand allows you to earn 0.5 CME credits per hour at your own pace, which, as you know, is incredibly valuable on its own. So click the button below to grab your VIP All Access Pass at a special limited time discount right now. Hello, and welcome to another Telomere Summit episode. I'm your host, Dr. Joseph Raphael. Today, I'm very pleased to have Robby Besner on to talk about photobiomodulation and its effects in health, to hear about his journey from helping his daughter with her illness through creating a world-class company delivering this therapy. Rob Besner, PSc.D, co-founder and science officer of TheraSage, has always been an advocate of natural health and wellness. Graduating from Boston University in pre-med, engineering, psychology, and business, he continued on to postgraduate work at Case Western Medical School and holistic medicine. After many years of illness, Mr. Besner's teenage daughter was diagnosed with Lyme disease. And I'm going to tell you, he's going to tell you his story about that just briefly in a minute. He is also a professor for the World Federation of Chinese Medicine Societies, an annual contributor at the World Committee on Infrared, a member of the Education Committee of the prestigious Hippocrates Health Institute, and a presenter at many medical, integrative, and anti-aging health conferences, as well as an invited contributor on many podcasts, online summits, on-air radio and TV health shows around the world. And he's also a best-selling author and considered a leading scientist in the field of integrated infrared technology. Robby continues to research and develop new device and health science applications to bring the power of healing with nature into the world. Welcome, Robby. I'm really pleased to have you on. And I always like to start by hearing the interesting stories that brought our interviewees to where they are in their lives at this point, and the technologies and science that they're doing.
Thank you for having me, Dr. Raphael. And I'm really, really very honored to be part of your summit and to have the opportunity to share my research and some of the great results that we've gotten in our clinic and you know, with your viewership. The backdrop, or basically the underlying story, the impetus of how we got started was somewhat serendipitous. In a sense, my oldest child, my daughter, contracted chronic Lyme disease in her early teens. And I'm going back about 30 years now. And 30 years ago, the amount of science and the advancements and applications in Lyme disease was just sort of scratching the surface in a way. So we didn't have a lot of resources in terms of Lyme literate doctors available and advanced methodologies and protocols at that moment in time. So having a medical background and other skill sets, it gave me the opportunity to kind of focus on my daughter's health and looking at taking a deeper dive into the ways that she was herself. And the way that the ones that became most apparent were high levels of inflammation and toxicity. And so when I kind of unpacked that a little bit, I focused on toxicity and the best methods and ways to detox. And so we know invasive forms like dialysis and certain kinds of IV therapies, and then less invasive ones where you're using either a pharmaceutical-based or natural homeopathic to stimulate some kind of chelating process to get toxins out of cells or tissues. And then I discovered that there are actually wavelengths of sunlight that can mobilize all the toxins in your body, and that really intrigued me. So as I was starting to look into that a little further, I realized that there really wasn't a lot of applications for the mainstream public to gather or gain devices and or protocols in that way. In fact, 30 years ago, infrared therapy and photobiomodulation was mostly used in the sports industry by sports medical doctors to kind of lower pain thresholds and improve or expedite the ability for the player to heal from an injury and get back on the field. And so all that stuff intrigued me, but how does that apply to me and my family crisis at that moment in time? So what I decided to do was organize a trip. I went over to Asia. I spent better part of 6 weeks. I interviewed more than 50 different factories and started developing devices, mostly for our home use. Our daughter experienced some really great results from these devices and applications, and her doctors, she had a team of doctors at that point, noticed that and asked that we start to develop these devices for their patients, because most of these practitioners their practices were almost 100% Lyme disease and co-infections that follow in the path, or they travel together as families of microorganisms. And so we started to develop these devices, and that was essentially the genesis of Therasage, the company. And so now, as time moved forward, obviously Lyme disease has advanced greatly, not just in the tri-state area around Lyme, Connecticut, which they consider ground zero, but it's fairly well spread across the US and has actually spread, I believe, in pandemic proportions around the globe.
Sure.
Mostly because we really don't have a statistical data to support a lot of the cases, because Lyme disease mimics many other kinds of infectious diseases. And so it's greatly misdiagnosed and obviously mistreated because of that. But bringing us back to the central point, it is clearly a problem. It's not the one that's in our face today, because we've got the pandemic and the COVID challenges that we're working with globally, but certainly it's in the backdrop, and it's one of the big ones up there, and one that I think needs a lot more attention to. So it is something that we do focus on here at Therasage and with my company, with my research. Now, what was so unique about what we found in terms of understanding light energy was that basically light is a wavelength and creates a frequency in the body. We call that bioresonance. Just like sound waves, light waves have a resonance in the body, and certain light waves actually resonate at the same frequencies that our bodies resonate at. And as I discovered and started to dive deep into the research on this, I realized that there were really many, almost a systematic approach that light energy can affect in creating what I call a primal healing platform in the body. And that's really where we have centered most of our research for the last close to 25 years in our company.
Wow. Yeah. So that is a really interesting story. You had a need, a loved one, and that created a passion that then you saw how effective it was. Let me ask you sort of a little bit of a primer on infrared. I mean, we hear about various technologies that are sort of non-medication ones. What's the basic physiology? We're talking about wavelengths, and you said it resonates with certain I guess, cells and mitochondria potentially. For our sort of, fairly educated listeners, how does that work?
Okay. Well, let me start with the sun generates frequencies, and there's a full range of them. They call it the electromagnetic radiation that comes from the sun, and it spans from gamma rays all the way to radio waves, being the least, let's say, the most benign on the body, infecting our health. Infrared spectrum sits sort of right in the middle, and it actually represents or is responsible for heating 82% of the Earth's surface. Infrared frequencies do not, they're a wavelength. They have 3 different sections, the near, mid, and far. Each of those wavelengths do different things to the body. That's what I found so intriguing. And as nature delivers these wavelengths to the Earth, actually some scientists like Nikola Tesla and Albert Einstein, they actually together did some work, but Einstein did a study in the early 40s that studied the effects of sun thermals on all life on the planet, and came out with fairly broad statements that without the infrared spectrum, there really wouldn't be life, both in the plant and animal kingdom as we know it today on the planet. So that's how important these wavelengths are in stimulating systematic primal healing in the body. They're responsible for structuring the water in the body. Well, why is that important? Because when we have structured water in the body, then we have better cellular communication, right? And also, having structured water is responsible for what's called protein folding. So we've got trillions of strands of proteins in the body that are looking to get sort of married together. And what the glue or the excipient that holds it together is actually a structured water molecule, a singlet structured water molecule. Absent infrared spectrum or that light frequency resonating in our body, that water would not be structured. These strands of proteins would not fold around, and we wouldn't have a full protein formed that the body can actually use, which is obviously, for people listening, one of the building blocks of primal health. So these are just some of the deeper science that infrared frequencies can do to help heal the body in many ways. And so today's topic is, telomeres and life extension or anti-aging. And there are multiple systems that infrared frequencies, the spectrum, the full spectrum, can influence in order to support the slowing of the aging process, and mostly just creating a really good health and wellness platform for longevity, or at least for as long as we're here, we're living in a healthy format.
So just a couple of terms that I probably should know, but structured water as opposed to unstructured water, what is that?
Okay, well, this is the body of work from Dr. Pawluk, who's probably considered the world's authority on water now. He wrote a book about 7 or 8 years ago that was revelating, called The Fourth Phase of Water. And in his studies, clinical studies, he noticed that the water inside the body acts differently than water outside. And as we all know, probably from high school days, water is either a solid, liquid, or gas, but actually the water inside of us acts differently, hence the fourth phase of water. Now, in order for a cell to actually methylate properly, we need to have structured water in the body. The water acts as a transport system. Structured water acts as a transport system to get nutrients into a cell and to allow the cell to get export toxins and waste products. So what Dr. Pawluk noticed in clinic was that all of his patients that he studied that had structured water, that had enough sunlight to have structured water in their body, they were basically healthy. Their cells were functioning properly. They had great vital signs and so forth. People that didn't get that sunlight, were absent that, they were more on the clinically sick diagnosed status. So it was very clear to him that light frequencies, and in particular infrared light frequencies were responsible for creating that polarity between sickness or disease and health simply by cell function. And you might agree that we probably get sick on a cell level and we also heal on a cell level.
Sure.
So everything that we can do to support cell health is going to cascade into general health, general tissue health, organ health, and systems health for the body.
So that's great. Yeah, that's a great explanation for the structured water. We're mostly talking therapeutic about near infrared, but there's also mid and far.
Yes.
And I guess we think about radiation as generally being bad. Gamma rays are not good. Radiation therapy fries things. This is non-thermal radiation, and it's generally really good. I mean, from what you're telling me, it's almost like we need it as much as we need oxygen, but maybe at a little slower. You're not going to die as quickly from it, but if you lived a mile underground for a long period of time, what you're telling me is you probably won't have as good health as if you got up to the surface more regularly. Am I correct in thinking that way about it?
Yes, and I'm not looking to go underground right away, but I think when you put it like that, yes, probably more obvious for people to know. And this is a little different range of frequencies. It's more appropriate for the UV spectrum, which is just before natural light, which is just before the infrared spectrum.
Right.
But people may relate to SADS, which is a condition of basically light deprivation that happens mostly around wintertime and or in the more northern areas of the world, whether it be US or European. And that light deprivation, what it does is it creates emotional distress, and also people, their bodies don't function as well during winter times. Well, what's happening? It's colder. You're wearing layers of clothes. Your skin, which is a photoreceptor, is getting less exposure to natural sunlight. Hence, all the things that we're going to be talking about today and my whole body of work slows down greatly during the winter times, which would suggest that having devices or ways to supplement, just like we do with a nutritional supplement, you can supplement with light. And in fact, in certain parts of the world, like the Baltic countries, which I've through my studies have come to understand, that culturally, many of these societies have embedded light frequencies and infrared sauna and saunas in general for both detoxing, general health and wellness, and to offset the deficiencies that they have in their part of the world because of the azimuth or the angle that the sun touches the earth at that particular time of the season for them. And so light is not well, it's not on the top hit list. Like you say, we won't live very long if we don't have water or we don't have oxygen, but no one really thinks that if we're light deprived, that in fact, it would create a physiological challenge for anyone. But in fact, it does. And it does move in a much slower pace, meaning the withdrawal from light frequencies will show its symptoms in a way a lot differently than they would from starving from food, water, or oxygen.
Yeah, that's interesting. That, I mean, it's known that higher latitudes have increased incidence of autoimmune disorders, infections, and chronic diseases. Some of that was thought to be from lack of vitamin D, but it's probably more than that, and quite likely is. So that's a really good explanation. From getting down to the cellular, sort of molecular level, how is it working? For instance, seasonal affective disorder is. I certainly understand that and want to get out of New York as soon as I can after the holidays. And I do use light therapy, so I think it's definitely a real thing. If anybody suffers from it, that's absolutely something important to do. But at a cellular level, the structured water makes things, I guess, function better. Is there more molecular detail that we know about in terms of epigenetics, gene expression, mitochondrial health that gives the benefit? And then, as a secondary question, how is that harness to deliver therapies?
Well, it's interesting because as we unpack the different wavelengths within the spectrum, so you have near, mid, and far. You referenced near infrared earlier. If for people that would be researching today, there's a big rage or a big divide between the effects of near and the effects of far. Of course, the providers that make far devices say they're the greatest. The ones that make the near say they're the greatest. The approach that I've taken is more of one following a page out of nature's playbook, because the sun delivers, or nature delivers, infrared in a full spectrum that's both near, mid, and far. And what we did, from a research point of view, NASA actually publishes the amount of these frequencies, which are measured in nanometers up until 1,000 nanometers, and then there turns into a micron. So the full spectrum of infrared is from 600 nanometers to almost 20,000 nanometers, or 20 microns. Now, that being said, these different wavelengths, it's almost like a FM station. They all dial in with specific frequencies or a specific nanometer frequency measured, and they all do different things. And so the near frequencies are principally responsible for things that are skin related. Skin and around 3 centimeters deep is the general penetration. But they also will stimulate mitochondria, which is the ATP, ADP cycle, circadian cycle, and also is the powerhouse of the cell. And so that's very related to anti-aging and to the length of the telomere, because if we can do something like bringing in near or infrared frequencies to the body on a regular basis, then we can extend the telomere length. We can support mitochondrial and cell energy, which really is a really important system in order for us to all be healthy and to support our immune systems and various different other systems. It all starts and comes from the cell. So the near frequencies do that. They also produce a natural transient gas, one of the 3 gases that the body makes, and it's called nitric oxide. Nitric oxide does 2 things. It'll create vasodilation, which increases metabolism, also something that is great for anti-aging, because the more we can move blood around, the more exposure to our tissues of whatever nutrients are in our blood, which are the DNA, RNA factors, as well as nutrients for supporting cell health and tissue health. And so increasing metabolic rate is very important, both on a micro level, which would be your capillaries, the smaller ones, which is more appropriate for the near frequencies, but it also triggers the hemoglobin, or your blood cell, to grab more oxygen when you make it available. So a wonderful synergy or stack would be to marry nitric oxide and more oxygen, like ozone therapy, for instance, or hyperbaric therapy. And so if you have near frequencies and or a nitric oxide supplement, and you combine that with an enhanced oxygen source, then you're really talking about changing the chemistry of your physiology on the inside, which obviously manifests in many different ways in helping the body keep a stable microbiome, for instance, which affects your GI tract, which also supports your immune system. I mean, there are cascades of influences that the area that we're working in can help to support good health.
Well, that's it. I mean, I've had a hyperbaric oxygen therapy expert on, and hydrogen water expert on, and various people that have these different modalities, and they seem to be, there may well be some significant synergies. Do you know of any papers looking at them working together? Has your company done any research on putting them together? Because it's something you certainly could do in the larger hyperbaric oxygen therapy chambers. But if you haven't, then the next question I have really is, you said potentially lengthening telomeres. Have you seen any actual lengthening of telomeres in studies? Telomere attrition slowing can happen from many different ways. Pretty much everything is good for you, slows down attrition of your telomeres, and that's because of reduced oxidative stress and more antioxidants around, et cetera, more mitochondria, better mitochondrial function. But have you seen actual increases? Because they have published that for hyperbaric oxygen therapy. One group in Israel published that recently. Have you seen any of that?
Well, we see a lot of studies in different areas, like for instance, redox signaling molecules. Many of these systems, just like nitric oxide and redox signaling molecules, as we start to age, and generally, I don't know why this is, but the precipice seems to be late 20s, early 30s, that at that point, as we're aging, we make less nitric oxide. Our body produces less of these redox molecules. And so that's that then, following that train of thought, we're getting more oxidative stress and we're getting less oxygen in our blood, or our blood's ability to hold on to oxygen. And this all contributes to our aging process. And so you can easily extrapolate that if you can supply more oxygen to the blood or you can supply more redox molecules as infrared frequencies do, and that kinds of science has been researched, has been proven in clinic, and also expresses in the way that patients respond to this kind of therapy in terms of healing themselves from potentially infectious or chronic ailments. And so they kind of all work hand in hand. And some of the specialists, and I'm very fortunate because in my small area, humbly said, I've come in contact with some of the greatest minds in the areas of research of nitric oxide. For instance, Dr. Nathan Bryan has a company, and he is the protege of the Nobel Prize winner who discovered nitric oxide. Personal friend, has developed supplements, and we actually stack and use these protocols with some of the devices that we've developed in our company. And so when we interview a patient and they present themselves with certain symptoms, and we see that, or we work with a doctor in their clinic, as we dialogue with them and get a better understanding of what they see in their clinic in their normal demographic, we can easily show them protocols that we've developed and the devices that we pair them with in our body of work that will enhance the efficacy of that platform or the system that doctor and or that clinic is treating with their demographic. And so we have seen in many areas, we use different kinds of testing equipment. Besides the electronic testing, we also use heart rate variability as a non-invasive way to understand exactly how the autonomic nervous system is affected by our body of work or the infrared frequencies. By the way, our approach has been a full spectrum approach. And so in nature, the sun delivers near, mid, and far in a certain proportion. It does change or vary almost second to second because our atmosphere becomes a lens or a filter that actually changes that cocktail. But what we did as a company on the research side was we took the first and second standard deviation of the data that NASA has produced to show us how much of these frequencies and in what proportion they come to the Earth on a daily basis. And that's been sort of our secret sauce behind the way that we develop our technology. That being said, 3 years ago, I have been teaching for more than 20 years at Hippocrates Health Institute here in West Palm Beach in South Florida. And I teach in a room that has a big mural of Hippocrates, and it was bothering me after like the first 18 months. And I thought to myself, one of his big sayings is food is your medicine, but the food of Hippocrates' day is much different than the food of today, right?
Right.
So is our environment. And so you mentioned epigenetics a little earlier. The way that our lifestyle choices and the environment that we're living in these days affects our DNA and RNA and the way that we express ourselves. And so I decided to shift a little bit from the playbook that nature gives us on a daily basis to really adjust to the times that we're living in. And in doing so, our latest iterations of our own research and development has been in devices that actually incorporate more of the near frequencies in proportion to than what was normally delivered by nature, we've actually dialed up more of the near frequencies to address mitochondria stress, oxygen problems, and so forth. And so I believe that we're living in a dynamic time, and we're also living in a very dynamic environment, of which our bodies are all, we're seeing it in every different way. Chronic disease is off the chart, the direction of the actual ascension of the growth of these various areas of the chronic disease areas are ascending, and they're not coming down with all the money and all the research that we're putting into this. And so I think through the evolution, we're not just seeing advancements, but we're also seeing in the advancements of the way that our environment is affecting and our choices are affecting our overall health. That's really what the deeper statistics are showing us. And what's really alarming to me is that the ages that people are being affected by their environment are coming down. Yeah. So we're seeing, and you may see this in your clinic as well, we're seeing, women in their early teens showing signs of infertility, men 18 or less that have low testosterone levels. This is not a genetic expression. No. This is more of an epigenetic expression. And I think we have real solutions that are not expensive, but it starts with us talking like today and creating more awareness as to the ways that you can stack different kinds of interventions to create a much greater result than just the single ones by themselves.
Yeah, that's very interesting, the work with Dr. Brian that you've done. I'm of course aware of him and use his products in my practice all the time, and myself. They work great to be able to couple that with the infrared therapy, full spectrum. I mean, when you're talking about how you've moved towards more of the near, even though that's not, that there's a full spectrum of infrared that comes through. We evolved as a species in an environment where we, reached our peak of health around age 25, and the vast majority of us were gone by age 45, certainly 50. Yes, some lived to be 50, 70, 80, but we're optimized for perhaps the needs of that full spectrum in those proportions when we're that age. But as we get older, we may need more of the near, and that's what your research is showing. And it's not, we're a species out of its natural environment. And so thinking that way, I think, is actually the right way to think about it, because, we don't move around as much. We have these decreases, probably more rapid decreases and longer time for decreases in NAD, the redox molecules, and nitric oxide to decline. So I think that's really fascinating. Let's talk a little bit about, I know there's conditions that can be treated with infrared therapy. The overall aging process, what would you say would be for somebody who's functioning, relatively well? We all have our strong systems and our weak systems, but what kinds of protocols, what kinds of things, and what kinds of devices, and then also what kinds of results should you expect for someone who wants to be proactive about their aging process, improve their performance, slow down their aging, and perhaps through all that affect their telomere length?
Well, that's a loaded question. We probably would need more than just today's interview.
Well, I mean, it's a start. Because I'm like, let's say I'm asking you as somebody that's thinking about, and I have many patients who are asking me, Dr. Raphael, there's a lot of devices out there. What should I be looking for? What's the best one? Are the ones you stand in front of, the ones you wear? What frequency? Sure. I know that there's obviously it's a hot area of controversy that you mentioned earlier, but some basics about what one might consider if one wants to avail. I mean, I know that one of my patients, I look at many, many biomarkers of aging from the vast majority of ones that you've seen in the literature. And one of my patients that's doing the best and actually reversing her aging from many markers is employing, fasting, PBM, PEMF, various other things. So she's on hormone therapy, but a lot of my patients are on hormone therapy. A lot of them are on telomerase activators. And so I think it's coming to sort of where there's a multimodality approach to this that I think is going to be really important. So I know it's a longer discussion. People can go to your website, I'm sure, to look at the options. But I mean, how often would you stand in front of a light or have one on you for general improvement of mitochondrial health? And then we can talk a little bit about skin. I was curious about that because I know when I go to my dermatologist, they have me stand in front of a light for a bit. And, usually with dermatologists, they're like vampires and don't stand in front of any light. So with that, maybe take a stab at it.
The science on light has changed quite a bit too. And I think before, well, I'm in South Florida, so here they want you to slap on these carcinogenic sun blockers, before you even step out and to get into any sunlight because of potential harm with carcinomas and various different skin related challenges or damage. I think the big ones in terms of my body of work would be probably the ones that affect aging in general, like better sleep, better diet, and toxicity is probably the biggest one. We're living in a toxic environment. People know that. When you can have a food source, and I'm not against Twinkies by any means, but when you can go to a gas station, buy a Twinkie that's got a shelf life of 500 years, you start to wonder exactly what actual natural nutrients are in that Twinkie. Okay, again, nothing against Twinkies. So, but in terms of toxicity, the body absorbs toxins in many ways, and for the most part, the ones that are environmental are the ones we will take in through basically water, food, and air, and absorb a lot through our skin. And the layer of fat just under our skin actually will hold close to 70% of our toxins. So in the detox process, one of the devices that we make in our company is an infrared sauna, for instance. And there are other sauna competitors and device developers out there, and they make pretty good devices. Again, we've taken a full spectrum approach, which is different than some of our competitors do, mostly again, because we're trying to harness nature's playbook. And in our belief system, there is something in full spectrum for everyone out there, because similar to vitamin C, the body will absorb what it needs, and what it doesn't, it just sort of spills out. Same thing with these light frequencies. You can't get too much of them. Whatever you need, or wherever the deficiencies are, the body will take that from the full spectrum when you offer that. And then whatever it doesn't need, it's just through the process of homeostasis will actually just let that frequency go. And so we got all the benefits without any of the drawback. It's quite revelating. Now, on the detox point of view, and the only caveat to this is, let's say for instance, infrared sauna, if we had an older person, an aged person that was presenting very fragilely, I wouldn't necessarily throw them in an infrared sauna and tell them to stay there for an hour, even though that's what they need. It wouldn't be where I would start. Similar to if I were a swimming instructor, I wouldn't throw you into the deep end of the pool without a life preserver. I'd probably start you off in the shallow end. If you were really presenting fragilely, then I may have just put your toe in the water where you got comfortable with the depth, we got comfortable with the temperature, and then gradually walk you into the deep end. So we are very conservative. We're a very low and slow process in our protocols, so that we are able to check in with the practitioner, the leading practitioner in mind, and also with the patient, making them aware of exactly what signs of detox and recovery they are going through, so that they aren't alarmed, number one, and secondly, that they can adjust accordingly. And so hydration is a big part of this. Earlier, we talked about light frequency structuring the water, but many of us, even today, we're a country that drinks a heck of a lot of water, but we are not cellular hydrating. And the reason why we're not cellular hydrating has to do with sunlight, because if we are deprived from these frequencies, then we're not structuring the water. So the cell isn't actually holding on to that water. And so it's not using it in the right way. In fact, the water is just passing through us. So that's important for us to know. And in order for us to detox properly, we have to be hydrated. And so that's in our protocol. But you asked a very important question. How long do you stay in front of the light? And so it really depends on your end game. What are you looking to try to achieve, right? Like in any kind of diagnosis and protocol. And so generally speaking, for the average adult, it takes close to 8 minutes to get enough infrared frequency in their body for there to start a physiological event. And so we look for a threshold, at least in the beginning, while we're acclimating, for something between 20 and 30 minutes at a very good heat or easy heat around 125 degrees Fahrenheit to 130, which is really just maybe like 22 to 24 degrees higher than your normal core temperature. So it's not enough to cause heat frustration like the other kinds of saunas per se, like a hot rock sauna or a steam room that are 185 to 200 degrees Fahrenheit, or some people have even trouble breathing in those hot environments. Where infrared sauna, you get absolutely just the opposite. If my schedule could permit, I would probably stay in it for hours on end because of the benefits. But again, I've been doing infrared sauna for close to 25 years, almost every day. And it is important to understand that this type of therapy or photobiomodulation is a cumulative kind of therapy. So the more you do it or more often you do it, let's say daily, and the time and temperature you do it will make all the difference in the benefit or the result that you get. So a great analogy is this, Dr. Raphael. Do you have any animals in your home? Do you have a dog or a pet of some?
Yeah,
some
a
sort?
cat. Mm-hmm. Cat.
Okay, so let me ask you, when your cat is comfortable in your home or your apartment and it's Sunday and you guys are just waking up, your family's waking up, and then you open up the shades, do you ever find your cat or your animal, and people out there listening could maybe be able to relate to this, they kind of meander from their pet bed or wherever they were comfortable, and they sort of move towards the window or the glass or the sliding door, and then they do one of these things where they kind of roll around and they kind of bask in the sun and so forth. And so can you relate to that?
Yeah, no question. They're always looking to find where the light is shining in, and they'll curl up there and they'll move with it.
Okay.
I definitely noticed that.
Let me ask you, did you teach your cat how to do that? Did you train your cat to do that?
Oh, no, of course not. They just, that's something they do.
So here we are, like a child, animal, they're showing us the playbook. Like here we are, the most intelligent high end of the pyramid, right? Yet we don't meander ourselves. We don't find ourselves going over to that same pane of glass, getting that sunlight. What they're doing is what nature gave them that information to do, which means we are light beings, all animals and plants on the planet. And so we need infrared spectrum every day in order for us to be healthy, a certain amount of it. Generally, it's somewhere between 20 and 30 minutes for the average adult. But the caveat is that in our busy lifestyles, we don't get out. If it's wintertime, we hardly get out at all, maybe, to get that sunlight. That's why we need to supplement or understand that in order for our body systems to function, our immune system to be supported, many of these, even on a cell level, mitochondrial health, natural production of antioxidants, things of that nature, we need this sunlight. We need at least 30 minutes of it every day. And so as a company, we developed techniques and devices and protocols that allow us to enjoy our lifestyle, busy lifestyles, and also get what we need from a physiological point of view to be healthy every day, at least to create that healthy platform.
So that's a really interesting anecdote about the cats and or animals in general. And I think you're absolutely right. I'm like that. I'm always looking for the sun. But in terms of just a little bit of confusion in my mind, and forgive me, I'm a neophyte at this. There's infrared light, and there are these things that you can stand in front of that don't necessarily heat you up. And then there's infrared sauna. I take it that you get the benefit of the infrared with these things that that can give you the frequency of light, but then there's just added benefits sitting in a sauna that also warms up as well, or for those that don't have the space or can't afford that, then standing in front of a light, or there's also ones that you drape on you. I know that for a while there, for my hair, you just put one of those caps on that gives you, that's I guess a little bit different, but it's essentially photobiomodulation as well. There's a difference between those two. The sauna part is additive to it, and you can still get quite a bit of benefit from, and it's the same amount of time, I guess, standing, and you have protocols and different devices for all that, or?
Sure. And let me explain to you, maybe we'll dive a little bit deeper on that because it's important for people to know. So there's basically two delivery systems. There's convection heat, and then there's radiant heat.
Right.
Infrared is radiant heat. that's light waves and a wavelength. So the sun delivers radiant heat, or these wavelengths, to the earth, and it does not heat up the ambient air, the atmosphere between the sun and the earth. It just shines on the earth, and the frequencies are absorbed in the earth, and in our bodies, they're absorbed in the densest parts of our bodies, which is our muscle mass or organ tissue. And so it, as opposed to a hot rock sauna or a steam room where you have an element that heats the ambient air up, and then that air touches your skin, and it has to warm your skin at a very high temperature in order for it to pierce through that insulating layer of fat under your skin that's designed by nature to protect our insides from extreme heat or extreme cold. And so infrared works differently because it actually resonates. Like earlier we talked about the frequencies. Well, there's a frequency of water inside of our body, and the infrared spectrum actually resonates at the same frequency as the water. And so almost 72% of a fat molecule is water. And so when an infrared ray or wavelength comes into the body, it actually resonates, called a bioresonance. It's a vibration. It penetrates right through that insulating layer of fat. So hence, we don't have to have a high elevated temperature in order to get all the benefits, right? And it now comes into the body and it's resonating, it's actually vibrating in an organ or a muscle tissue, which are the most vascular parts of the body. And so when that vibration actually liberates heat, warms up that area, warms up the vessel, creates vasodilation, now we're increasing metabolic rate, your blood flow, by close to 15, almost 20%. And all the attributes that go to that: better oxygenation, better nutrition, or potentiating nutrition by your tissues and organs and so forth, cells of the body. And so all of these functions are all circled around these wavelengths. And so, yeah, they work in that way.
So the sauna then sort of just combines them when you're in the box, combines it's warm inside there as well. And then there's also the infrared. So it's a combination of those two. When you're standing in front of the light, do you feel warm? Not on your skin, I guess, but you feel warm inside. You start feeling warmer inside you. Okay. And it doesn't matter whether you said 3 centimeters deep, or is your full spectrum go deeper?
The near frequencies will penetrate 3 centimeters. The far frequencies will go somewhere between 6 and 8 inches, depending on the density or the BMI of the patient as they present themselves. And so that varies from person to person, but generally that can be adjusted by time and temperature. And the temperature will promote sweating, which is one of your best detox pathways. If there were only one thing we could talk about, that would be today to extend the aging, slow down the aging process or extend your life. I'd have to say, if given today's environment, if we could lower your toxic burden on your body, which also lowers inflammation, and keep your body hydrated, we're going to improve your overall profile greatly. And so that's the big message. And infrared sauna, using it regularly, will help detox the body, keep your detox pathways open, help support your immune system. It will also affect your autonomic nervous system, shift you from sympathetic dominance, that fight and flight, very anxious feeling, to being less stressed, more relaxed. We all know from therapy and clinic that when your body is in parasympathetic mode, that your body will receive almost any therapy and embrace it in a much greater and more sustainable way. And so by simply going in infrared sauna, and we in our protocols, we suggest that you follow nature's playbook and go in the earlier part of the day, almost when you wake up, similar to the way our ancestors, way back when, 8,000 years ago, we lived on the land, we lived in tents and caves, and we woke up to sunlight. It woke up our body systems. This is the power of what we're talking about when we harnessing natural sunlight, and particularly the infrared frequencies.
Wow. So that's sounds like it needs to be done pretty frequently. In terms of, I'm just curious about, I know there's lots of studies looking at applications and various conditions. Are there any hard endpoint studies looking at sort of markers of the aging process and, say, a 6-month course of or even 8-week course of infrared therapy on, you name it, some process of the aging process. I'm not familiar with the literature.
Well, again, we've taken a more systematic approach, and in all of our work, we have proven our own work in clinical applications, but we back it up by PubMed and various different articles and research and assays from the New England Journal. There are many, many publications that we can reference, and I'm very happy to supply that to you in any area that you feel comfortable to give to the audience, so that they can feel comfortable researching and understanding exactly how deep the science is on my area of my body at work. So yes, and we're happy to do that. The other area that I think is really important, and it really comes under the bigger category of stress, which also affects our aging process. And today we have another stressor out there, a more modern one, which is the effects of the exogenous frequencies that we're being influenced by, like electromagnetic fields. And now the newer one that we've been doing a lot of research on, which is extremely low frequencies, coined as dirty electricity, they call it. But all of those man-made frequencies do affect us. So today we've been talking about a whole bunch of frequencies, sound frequencies, light frequencies. Well, there are these man-made frequencies, and EMF, 3G, 4G, now 5Gs, the government, our government, and the military are actually posting 7 and 10G amplifiers on the same microtowers that they're delivering our phone systems to. So the challenge here, Doc, is that we can't see them, hear them, smell them, taste them. And so, but we can see certain ways that it does affect us. Like, for instance, there's a practitioner in the Pacific Northwest named Dr. Dietrich Klinghardt, and he's done a blood study where he studies the effects of microbial activity in the blood, meaning that a microbe, a parasite, a mold, they will, in their life cycle, they basically die. That's a toxin. They have biofilms that surround and protect them. And they also poop, which is either a neurotoxin or a microtoxin that can be measured in a blood study. And so what Dr. Klinghardt did was he took 200 patients, half of them, or 100, were from the urban areas and 100 were from the rural areas, all basically drinking the same water, breathing the same air, and eating the same food. But the difference, dramatic difference, is the concentration of microwave towers and cell towers in the cities versus the rural areas. Well, it turns out that when we measured the microtoxins, there was 85 to 90% higher levels of microtoxins and neurotoxins in the blood serums of the people in the cities. And the only way we can explain it is that these exogenous frequencies are actually energizing the bugs, and the bugs are replicating, they're using more of your body resources, and they're pooping more.
Wow.
So this is fairly significant on a stressor that is more modern, but still affects our aging process greatly, because stress is a big one. So infrared frequencies will pull the EMF that the body is holding. We as a company started studying and remediating EMF almost 17 years ago. So we are very advanced in our own applications in the way we remediate, so that when you're sitting in one or using one of our devices, you're getting the pure frequency that, as the sun delivers it, not all the man-made effects like the electronics and the EMFs that might come from the device itself. And so you're getting all the therapy without any of the full drawback. And I think this is important for people to understand, that the body is affected by all these stressors, and of course, ultimately it rolls up to affecting us in our aging process, or our ability to maintain good, vibrant health to almost our last breath.
Well, that's a really, I think, a good way to close our discussion, unless you have some final words you'd like to say. This has been a really fascinating conversation, an area that I definitely want to learn more about. Having lived in New York City for the past 25 years, bathed in all sorts of electromagnetic and other kinds of toxins and pollutants, it's time for me to do more detoxing. So do you have any parting words? And certainly you can just say your website is Therasage.com, is that correct?
Yeah, that is. You can get to us by our 800 number, which is 888-416-4441. You can email us at info at Therasage, THERASAGE.com, or go to our website. We also are public on Instagram and Facebook and so forth, so you can contact us that way. In parting, we're living in a sort of a tumultuous terrain these days, both environmentally, from socially, politically, all that stuff. It seems to be hitting all at once. I think that you can easily be overwhelmed. We have a tendency as practitioners to focus on and compartmentalizing the challenges that as the patient presents themselves. I think it'd be nice to take a little bit of a step back and look at things more holistically and more systematically, that how things affect each other. And I'm happy to be here today to at least introduce the idea that you can harness sun energy, as we have in various different devices and applications, and the appropriate protocols to improve the efficacy of whatever the doctors are doing that are listening today are doing currently in their clinic. There's something in it for almost everyone. And so I love that, because we don't believe that our position and our body of work is instead of any practitioner. It's really, we work hand in hand with the practitioner and the patient to try to improve health and general awareness. And it starts with education. And that's why today's session and today's interview is so important, is to just introduce the idea that there are actual modalities and interventions out there that are affordable, that you can easily integrate with your current protocols, that will make a marginal, a magnificent difference in the end result. And we both know that oftentimes just the order that we do a protocol in or the way that we dose a protocol can make all the difference in a moderate result or a fantastic result. And so let's go for fantastic, right?
Yeah,
and,
absolutely.
And I'm all for it. We're all in. We're ready and available, and we work hand in hand with the practitioner. And I'm very, very grateful for you asking me to be on this interview and to join your summit today.
Well, thank you very much again. And I look forward to running into you perhaps down on the beach in Boca sometime soon.
Come on over. The water's fine.
All right. Take care. So there you have it. This wraps up day one of the Telomere Summit, and I hope you've learned as much as I have. And make sure you tune in tomorrow, because we have another set of amazing speakers ready to share their expert insights and breakthroughs on not just using telomeres effectively in your own practice, but also educating your patients to help them live happier, healthier lives. But if for any reason you feel you might miss any of these important interviews during the summit, I do encourage you to secure lifetime on-demand access by purchasing a VIP all-access pass today. That way, you won't miss anything, and you can watch, listen, and take notes from these interviews again and again, and even earn up to 0.5 CME credit hours each hour, all at your own pace. Simply click the button below to order your VIP all-access pass at a special limited time discount right now. And before we wrap up, let me know what inspired you most about today's interviews in the comments below, and how you plan to implement what you've learned into your own practice. Thank you once again for joining us for the Telomere Summit, and please remember to share this with your colleagues so we can all work together to make our industry better for everyone.